Thanks for coming. Welcome to GenoFest 10. It's been a long time since we've seen people in this room. It's exciting. I hope that there are also people on Zoom out there, and that I think for a lot of people this might be the format that they'll consume GenoFest in the future. So I have the privilege of starting off, and there's three big things I want to talk about today: big iron, big changes in big data. But first, a little bit of an intro, and then I'll have some closing statements too. Let's start with the intro. This is our 10th GenoFest. Since 2011, we've had a number of GenoFest events, and as is usually the case, that we have some kind of a focused topic. So the event was called IllumaFest originally because it heralded the arrival of the first next generation, well, short read next generation sequencer. We had one next generation sequencer before from Roche, but when we got the Illumina sequencer, the GA2X in 2011, we had IllumaFest, and since then we've changed the name to GenoFest because the event now is about more than just Illumina and sequencers. It's really the whole ecosystem of different folks. So I'm not going to read all these out for you, but you can see that over time, as we've introduced long reads and single cell microbiome focused on communications, each year we've tried to bring something new in. Of course, then COVID hit, and for a couple of years, we didn't have GenoFest at all. And so today is really, rather than having a specific technology theme, it's really just to kind of reset. It's just an opportunity for you to meet us again. And so it's our presentations, and then we've got 3 presentations from some of our major vendors in the afternoon. Rule number 1 of GenoFest is to meet us. It's an opportunity for us to get to know you. That's why we like people to be here in person when possible. So let me just briefly show our org chart. One change is, I guess you know me, I'm the director. We've also created a position for Denisha Wallach, who I'm sure most of you know, who is now managing director, and she's overseeing all of what we call core operations, which includes 5 of our ops managers and then the folks who work under them. And so these are the folks who do all of your projects in the lab. In addition, we've got support folks, of which I'm one, also communications and finance folks. We've got an informatics group that keep the pipes open between us and MSI and provide primary data analysis, operations R&D team who work on new technologies that are going into standard workflows, and the innovation lab. And you'll hear more from various people today. Daryl's going to talk, as well as John and Fernanda, Aaron and Elyse. And so they'll talk about various aspects of operations and informatics and the innovation lab. The other important thing at GenoFest is to meet the vendors. They're out in the hall. We're always thankful for them coming. We're also thankful to 3 of the vendors for helping support GenoFest, pay for food and so on. They're speaking this afternoon. So we encourage you to go and take advantage of their investment in coming here to learn more about them. Okay, I want to talk a little bit about the evolution of life, of genomics and life science technologies. And this is a plot. The y-axis is PubMed citations for Southern blotting, invented in the 70s by Ed Southern at Oxford. Northern blotting, cheekily named after that for DNA and RNA profiling. And as you can see, it really looks like evolution of an organism, a growth and a crash. And if you plot those both against the normalized max, you can see Southern blotting was invented earlier. It crested and rose and then crashed, so did Northern. And of course, the genomics era probably had a lot to do with that. These were kind of first generation molecular biological techniques for looking at one thing at a time, or very gross structural rearrangements, restriction enzyme fragments. I mean, it was very slow and piecemeal. And to show you that this isn't just that blotting somehow became unpopular, this is Western blotting, our cousins in proteomics. And as you can see, whereas both Northern and Southern blotting kind of went away as the genomics era came in, Western blotting is still going strong. And the reason is because nucleic acids are a little bit like digital data. They're very amenable to being processed all in the same way. Every protein is, of course, its own animal, tricky. Proteomics doesn't work as well. It's not to put proteomics down, but the point is it hasn't scaled because it's not inherently digital the way DNA and nucleic acids are, and it's not chemically uniform the way DNA and RNA are. So a lot of the power of what's going on in genomics is because of this. So let's look at the evolution of genomics in the UMGC over this time. If you look at the term capillary sequencing, so the very earliest sequencers that the UMGC had was a capillary sequencer of the type that was used to sequence the human genome. And you can see that it rose and crested. And in the first decade, these were both technologies that we had in place. In fact, we just shut down the Sanger sequencing this summer. The folks made their own arrays. We had Illumina array systems. Genotyping refers to, I mean, you can still do genotyping with sequencing, but this refers specifically to targeted approaches that were much more important before you could do whole genome sequencing. And that too has kind of crested. That was the first decade of the UMGC. The decade from about 2011 to 2021, say, is the rise of next-gen sequencing. And you can see that it's just continuing to grow, but it's not a new technology anymore. And we've been in it we probably timed our entry into that just right, not too early, not too late. What's interesting too is you can see that RNA-seq, which is the next-gen approach to doing gene expression analysis, clearly is responsible for killing northern blotting. And microarrays, because microarrays really became, they were a tricky quantitative way to do what you can do digitally with next-gen sequencing much better. Long-read sequencing, of course, has been around for a while, but it's really come to the fore in the last 5 years. This isn't a case where we might have actually gotten into it a little early. We had a lot of bleeding in those early days because it was very tricky, but it's taking off. So we do this. It's a useful way to kind of keep up and make some predictions of what's going on. So the first decade were these technologies I've described. The second decade were those technologies. What about the next 10 years? I think that, as I'm going to mention today, with an advent of an incredible array of new sequencing technologies and ancillary technologies that support and lead into next-gen sequencing, this is going to be about whole genome sequencing, single-cell work, spatial work, counting applications that require tens of billions of reads, and an incredible proliferation of technology. So the genomics revolution is nowhere near over, but it's definitely entered a new era of maturity. The other thing that's going to happen is the sequencer smackdown. So those three players, we've got all three of those types of instruments, and you're going to hear from those today, but they're all of these new players, and that's going to actually really change things. The other thing I want to do today in the intro is to shamelessly pander to my new boss, Shashank Priya. I don't know if you want to wave. We're thankful that Dr. Priya came today, and a lot of my talk today, if you're hearing things you've heard before, I've pitched about have this talk to him. It's an intro to him. He's a busy guy. I don't necessarily get FaceTime that often, so I have it now. Okay, three big things. Big iron. That's the definition of the slang term of big iron. It refers to powerful computers such as mainframes. And that's, I think, agate or Masabi. It's one of the pictures from MSI's website of what they run over there. And as you know, Minnesota was a storied place in supercomputing, had one of the first supercomputers in the world. I'd like to start to think about us as the MSI of life sciences, and I'm going to keep saying that. And in a way, it's because what we're doing is we have big iron chips. All right, that's kind of the point I want to make. This is a picture of big iron we don't have yet. It's the new NovaSeq X. The timing with GenoFest X was coincidental. But this would be 4 of them. So that you're looking at over $5 million worth of sequencer there. But if you look at what MSI does, it surrounds its hardware with this infrastructure: people, physical plant, expertise for running it and fixing it, and consulting on it, and so on, and it results in outputs. That's exactly the same thing that we do. We also have hardware, physical plant, core operations, consulting expertise, have to store the data and the reagents in our case, and that gives you analysis. So in many ways, we're cousins, MSI and UMGC. We do a very similar thing. We both have to spend millions of dollars on instruments that last 3 to 5 years before they're obsolete. They're digital in different ways, different number of bits. But we're also partners, of course, right? It's not like we're separate. In some ways, we really work together to convert, the raw material of biology into biological understanding. And actually, that's what you do using these two resources. As I'll say at the end, despite the fact that there are these parallels, in fiscal terms, we're treated very differently. So the other thing I want to point out is that we're actually now literally big iron, potentially. The UMGC, if it gets the NovaSeq X, this box that I'll talk about later, It actually has got a supercomputer inside it, which explains partly its $1.3 million price tag. But it can actually spit out data itself. And so it's even possible that we will literally be big iron. I wanted to also briefly point out that although the UMGC is largely infrastructure, we also have a scholarship component. We're an academic unit. And that you'll hear about today from Daryl Gohl. There's tremendous synergies between the part of our lab that the part of the UMGC that carries out its own grant-funded research independently, and the UMGC's operations. So the two are, as in most genome centers, important and synergistic. Okay, what's our big iron? Well, that's our current suite of boxes. As you can see, largely they're from Illumina. And the reason is because Illumina really has become, I don't know if some of you are too young to know the term Wintel. So it was this Windows and Intel combination that dominated computers in the 90s. And Illumina is really the Wintel of sequencers. They weren't the first, but they did a much better job with a few technologies, a way of amplifying DNA on flat surfaces to get it to about 1,000 molecules of a clone that would give you enough boosted amplification to then analyze that data with what's called sequencing by synthesis, or SBS chemistry, another innovation in enzymology and chemistry. And then they had great optics. And this was massively parallel sequencing. What they got was a degree of scale and output that was revolutionary for all sorts of applications. They're also extremely good as a company in terms of innovating quickly. So they continued to consistently improve their instruments. They came up with things called pattern flow cells. All the sequencers right now have got little wells in which reactions occur. Two-color sequencing by synthesis that doubled the output, blah, blah, blah. But since 2017, they've been pretty quiet. There are also two kind of Apple company-like sequencer outfits out there, PacBio and Oxford Nanopore. They've made huge advances, and they've done it by being orthogonal. They are in many ways the un-Illumina. They generate extremely long reads that are orders of magnitude longer. They also generate orders of magnitude fewer of them. And so, they have incredible technology. They have a very devoted following. They can do unique things you can't do on the Illumina instrument. And so that's really been the landscape. But now, with an event just a couple of weeks ago, there's been the introduction of something called the NovaSeq X. It's the long-awaited new big deal from Illumina that everybody knew was going to come. And the event was very much like an Apple event. I mean, they had, the guy with the British accent, the screen, the whole thing, hushed tones. It was a little much. But, perhaps it was justified because it's an incredible instrument. They've got a new chemistry that's more powerful. And that chemistry, which will be available, by the way, on our NovaSeq 2000, is going to be the primary foundation for the NovaSeq X. It's got all sorts of new technologies that ultimately come down to this bottom line: a $200 complete human genome. It's not quite the $100 genome that others have claimed, but that's pretty damn good. It also has the computer on board that can pump out analysis. You could even basically, at some point in the future, discard everything except for your analysis. That might be where we're going, because the storage may be not something you want to do, given the tremendous output of this machine. It demultiplexes in real time. It's got reduced data storage. It could do 20,000 human genomes per instrument per year. And it's very much focused on human genomes. At least the presentation was. They've also come out with some long read tech information, and come to the talk this afternoon to hear more about it. I'm not going to go into it. They're the experts. It's very exciting. However, it looks like they're not alone. And this is San Diego. It's become like the sequencer hotbed, probably because the people from Illumina, when they left to form these companies, didn't have to drive far. Why did this happen? Well, Illumina patents expired. A lot of scientists who were vested left Illumina to form new companies. And of course, Illumina established that this is a really, really lucrative market. What it means to us is that it's a buyer's market. And so I'm just telling you all now that we will all together as a university decide what comes next, but we're going to take our time, because it's a time of a lot of ferment, and you don't want to be making a mistake now. This has happened in the past. We were very lucky in not making any bad sequencer purchases. We made some other bad purchases on littler things, but not sequencers, and so we don't want to do it this time. And then, of course, some are up in the Bay Area. Of these instruments, how have they done it? Some are Me Too boxes that are very, very similar to the Illumina instrument, but somehow skirt patent, or now that patents are expired, they don't have to worry about it. Some of them are out of the box. A company called Ultima has come up with a really kind of completely wacky way to do things that's exciting. And it's not over. PacBio itself, the long read company, they're now making a short read instrument too that sounds pretty intriguing. So, and there will be others. So we're going to get a NovaSeq, right? Well, not necessarily. It's a $1.3 million instrument. And with maintenance contracts estimated, we're talking about a $2 million investment over 5 years. I don't know if you realize this, some of you, but we don't actually even own our current NovaSeq at Fairview's. It belongs to Fairview. We just rent it. It might be hard to assess lower NovaSeq costs because it's such a large instrument, equivalent to about 3 NovaSeq 6000s. It might be the kind of thing we can only fill once every 4 to 6 weeks to run, which of course wouldn't be ideal if you want your data back quickly. Although they will have smaller flow cells, those won't necessarily be at that really low price point. So these are all the factors we have to take into account. It's not just cost that matters, but also things like, what are the configurations of flow cells? What's the turnaround time? And not the turnaround time of the sequencer, but turnaround time to fill the thing, get it full so it's efficient, and run it. What about things like backwards compatibility for people who've already got a lot of data on one platform? What about accuracy and required depth? Read flavors. You have a lot of short reads, long reads, different size flow cells. Reliability. You don't want to get into a platform that's going to go away. The ecosystem in support from vendors. A very interesting thing is happening where some of these vendors are providing their own chemistries, but others are saying, we're just the sequencer. We'll work with anyone, which also is analogous to the PC industry. So there's a lot of stuff to decide, and I'm announcing Operation Next Gen. We will have meetings in November, December, January, February, continue these, try to time them with events that are being held by these other companies, and be very, very open about the decision making, so that the money that we do have, we spend correctly. Lastly, there is some big iron in sequencers that really won't be part of our stable. When as a sequencer, not a sequencer, well, a lot of these new spatial in situ platforms, for instance, the Cosmics and Xenium, they sequence right on a tissue. And honestly, they're much more like microscopes than they are like sequencers. It doesn't play to our expertise. It plays to the expertise of places like the Imaging Center. And they're probably the folks who will have those. Okay, let's move on to big changes. As you may know, we got rid of Sanger, and we no longer help provide oligos. And I think the community was very kind about the fact that we're getting rid of these. We had to. They're very old technologies, and we need to make room for new stuff. We also got rid of labs. That's what we used to look like and what we look like now on campus, although we've actually increased in square footage because we moved about half of our staff 10 miles away from the university east to Oakdale. And the reason for this is that, as you can see on the left, we're really trying to focus everything on campus on our internal sales organization functions for you, and as much as possible moved out to the to forefront external services, also high throughput instruments and workflows where we've got a lot of space. It's a beautiful building, wonderful labs. It's like a whole wing of this building, and it's cheap. The rationale, I think I'll go through these quickly. The bold headings here is that we need surpluses, which we generate from our ESO, to help support basic fixed costs. Internal services were crowding out ESO, like when we really hit something big with ESO services, we really didn't have room to do well with them on campus because we didn't have room to expand. And similarly, the external services were getting in the way of ISO. So this segregation, it really helps with efficiency. And also, there's nothing special about UMGC space versus other commercial space. We pay for it one way or another. So, you get the best space you can. And that was out in Oakdale. So this is kind of a breakdown of what we do there. And you can see that on campus, we've got those different services in the 2 sites. So in this building is our big next-gen sequencers. And then out at Forefront, we've got these more high throughput workflows, extraction, array genotyping, array methylation, a lot of COVID work. Most of these are things that aren't used that much internally. Many of them support other Minnesota institutions like the Minnesota Department of Health, contracts with the CDC, wastewater testing for the Met Council, and so on. So we really are happy with this division of labor, and we hope it's going to work well. Let me talk a little bit about custom services. And these arrows show you services that are now going to be provided in that format. This is from way back. I'm sure most of you have seen this before. It illustrates that next-gen sequencing, which is this can in the middle of the kind of inputs and outputs. It's the engine, very much like a computer, that connects different workflows on the left in the lab to different informatic pipelines on the right, and means that next-gen sequencing can be used for a huge number of things. And of course, the last 15 years now have been all about the unending proliferation of these different techniques. The creativity of scientists has been overwhelming. Illumina, I don't know if you still have this, it used to have a great poster called "For All You Seek," just kind of put in one place all the different workflows that we had at the time that were out there, the ABCs of NGS, as we call them. This is just a blow up of one small area. Any of these could be things that PIs here at the university would use. And of course, it's impossible for any core facility, let alone one our size, to just master all of those. It can take weeks just to learn one of them. So the point of custom services is to allow us to access those. There's an added complication in a lot of new workflows like spatial and single cell, in that, as you can see here, for a single cell experiment, you've got the PI from working in a clinic or in their own lab, having to work with the flow core facility or do their own cell prep in their lab. They then give material to us, and ultimately it goes to MSI. So these workflows have become incredibly complicated, very powerful too. The challenges from some of these are, for instance, the input is now not DNA being dropped off or RNA being dropped off, but living cells that have got a shelf life that need to be worked on when they're ready, and people might not know when they'll be ready. It might be at 3 when they say they'll be ready. It could be at 7 because it took time to get them done. So you've got all these scheduling problems. The solutions, really, this is kind of specific to single cell, but the solutions are to have a more custom type of approach. And I think I'm going to skip over a lot of these details, but the point I'm trying to make here is that single cell genomics is particularly challenging because of this. And so it's going to be, a new challenge to really do it well for us. We're looking at automation, but a custom approach is a good one. So having automation in place that allows people to do it themselves, training people to do some of these upfront workflows, and then if people want, being able to use this custom model. This is exactly the same with our spatial platform. So the NanoString Genomics, NanoString's here today. This was purchased through a grant from that went to Laura and Paul in the IBAM group. And we also got some help from the Masonic Cancer Center to support both Fernanda and Grant in the UMGC and UIC. You'll hear from Fernanda today, who provide subsidized custom work on the spatial platform, which, like single cell, is complicated. Okay. Let me talk about big data. So this is a slide that from way back. If some of you have seen it before, I apologize, but I really wanted to make this point for Shashank, Dr. Priya. Many of you are probably, you're familiar with these companies, but you may not know what they refer to. That's the acronym for Yahoo, yet another hierarchical efficient oracle. It was a name that they came up with to describe what they did. And just in case you don't realize that, for a while there, Yahoo had an army of like hundreds or thousands of people who would just literally manually curate the web and put it into an index. I mean, that's what they did. That was like their business model. It seems ridiculous now, but of course, they were one of the biggest things in the internet at that time. And what they were doing was interacting with information in the way that people always have for centuries, which is that we categorize things, we group them down. That's how you do it. You study subjects separated, you study sub-subjects, chemistry, then you get biochemistry, organic chemistry, and enzymology, and so on, right? Google, of course, their approach was totally different, and their name reflects 1 with 100 zeros. That's the number Google. And their approach was no human intervention and algorithms that bottoms up via relationships between websites came up with algorithms that self-organize the web. So that difference between Yahoo and Google, because Yahoo is dead, right? And Google's a behemoth, is that this human annotation and hierarchical ontologies, which is what Yahoo even stood for in their name, it's clear that computer algorithms and networks are much more powerful in the modern big data world. So that was that message. Well, simultaneously, this is something biology's been doing too. If you look at the way that the Human Genome Project was organized and executed, literally, it was like the Encyclopedia of Life was broken up into chromosomes, and people got chromosomes. One center would get a certain chromosome, another would get a different set of chromosomes. It was cloned into large fragments, kind of like books, and then those were broken into cosmids, and those were broken into plasmids, and these were tracked, like literally, that's plasmid 1's got plasmid 1.1 and 1.2 and 1.3, and freezers and freezers of stuff being manually tracked, right? And meanwhile, over at Celera, Craig Venter and his renegades said, screw that, let's just shear the whole thing. We're not going to keep track of anything. We'll just randomly sequence what comes out of it. They were not doing this with the next-gen sequencer, mind you. They didn't have those then. But still the approach was you don't actually have to keep track of things with humans. And it's not fair to say that that approach worked and that the Human Genome Project didn't. There's a lot of debate out there. People claim that if the scaffolds from the human genome, kind of more careful Yahoo-like approach, weren't available because they were public, Celera could never have assembled their genome. But I think in the long run, this idea that random sequencing and saving all the time up front in not trying to organize information in a hierarchy has won out. And in fact, that's what massively parallel sequencing is. You take material, and it's coming from any number of places, even single cell is similar, and you use tricky ways to be able to keep track of it, but ultimately you use assembly and mapping and modern technologies to figure out what those are after you've got the data, as opposed to trying to organize it ahead of time. That's a picture of a flow cell. So, in other words, it's about scale, right? It's about that scale is something that biologists have had to learn, along with the rest of the world in our daily lives, over the last 20, 30 years. The world's awash in big data. And actually, what's interesting about genomics is that there's two different dimensions of this, which both live in the UMGC. There's the full complement of tens of thousands of genes, or the full complement of whatever your cohort is. It could be tens of thousands of people, or the entire human population being sequenced, or something like that, right? So the technologies we've put in place, these are just those for gene expression, have always had to try to capture both of those axes. So, and right now, for instance, the forefront external services technologies tend to be more focused towards this end of things. We've got more technologies for up here, lots of samples, smaller number of analytes. And then there's, of course, just a small number of samples, but with whole genome. And of course, up here is where, if you're an enormous center, you can kind of play with both. But we always have to have technologies in all these areas. So similarly for genotyping, a lot of these technologies are still around, some aren't. We've always had to cover this space. Same thing for sequencing. We have different sequencers for different scales of analysis, right? But in the last 5 years, a third dimension's been added. And that third dimension is that in most of those cases, you're still treating DNA as DNA, it's liver DNA, or it's a person's DNA. But now you've got the fact that you're talking about perhaps tens to hundreds of thousands of individual cells. And of course, we are made up of many more of those. Genomics, the bottom line is, just ignored biology for a while. The same way molecular biology did too. And that was very productive. It's not a mistake. But now that the technologies allow you to go back and look at them, I think what's becoming clear is one must. Because what do you understand if you're treating liver as if it were just a thing? I mean, liver isn't a thing, at a genomic level, it's just a combination of a lot of different cells that we call the liver in a very Yahoo-like way. But bottom up, who knows what it is, right? Some of the cells in there are much similar to cells in other organs, right? So the problem is when you start to do this, this is what you get. I'm sad to say that I don't think in my career I'll ever really understand these t-SNE plots and UMAPs. The level of abstraction that is now required to understand data is at the next level. And the reason I point this out is because it's a challenge. I think we find our own clients often saying to us, we recently heard, can you make us a UMAP? And I mean, of course, you can always just turn a switch and run a machine, but these are not obviously comprehensible, right? And even small, you can see over here on the left, this shows the effect of different parameters on this t-SNE plot generation. So this is a really big challenge. One of the things we're going to try to do is John Garbe is looking to hire another person because there's certainly we're not informaticians per se. That's MSI's job, but there are certain canned reports we can run, and so we're going to play around with this idea that we might be able to get people some of the way there by running some of these canned analyses. John already does this for single cell. He runs a lot of tools, and we can make that either part of our standard offering or perhaps something we charge for separately. And again, we have a great relationship with the rest and MSI. We'd never step on their toes. But to the extent that this might actually help them too, we'd go ahead and do it. Okay, Steve Jobs had this nice term, a bicycle for the mind. And that's how he described computers. And if we now think not about us as researchers, but about end consumers of genomics, like a doctor or somebody in agriculture, say, you're not, they don't want that for sure, right? They want something else. And those something else's are dashboards of some sort that convert, that translate some genomic information into something that makes sense in their terms. And this is true in the clinic. It's going to be true in a lot of places. And I would actually argue it's even true for you as researchers a lot of the time. So for instance, the QC reports that we generate are dashboards that John will probably show you this afternoon, Garbe. So here's a health dashboard. This isn't genomic information per se, but it is probably where we need to go. Similarly, in agriculture, here's just another dashboard that shows that you can do a microbiome analysis, but ultimately what the farmer wants to know is, do I put on fertilizer? Do I have a problem? And so on. I also want to make another point. Here's a screenshot that it's always embarrassing when my wife can see that I spend money in the candy machine. But what's remarkable about modern IT systems is their incredible comprehensiveness, right? I mean, you don't expect to be like, oh, that just failed to hit my Visa account. No, it doesn't get lost somehow, right? So the level at which IT runs in our economy right now is extraordinary, right? We take it for granted. And that doesn't exist in genomics yet, right? Although Daryl will talk about ways in which he is working on that. So this is an example of a Genome in a Bottle consortium from the National Institutes of Standards and Technology. They're realizing that we're getting all this big data, but it does not have data quality. And so they're developing standards. Here's an example of one nice approach. This isn't from us, but you can take a sequence and you can reverse it chemically. So instead of being 5' to 3' here, 4 A's and C's, G's, and T's, you make T, G, C, A, right? Chemically, it's exactly the same, but of course, in informatic terms, it's completely different. But the thought is that in terms of secondary structure and so on, it'll behave just like the original molecule. But when you sequence it, you will never confuse the two. And so what they showed is that with these sequences, you can spike them in. And as you can see here on the right, after you do library creation, you get an imbalance. Those of you who do library creation with us know that that's the case, but you can correct for it with these. So actually, the UMGC, Daryl and I, and the Innovation Lab is continuing this work, have patents for such spike-ins. They were, in fact, patents that were licensed by the spinoff company that the UMGC created some years ago. And Daryl's continuing to do really cool work on these kinds of metrics to make the data better. Okay, let me close with a little bit. That's the UMGC staff, and please meet them. Those are the staff who are supported by the university with any kind of funding, so none. our budget right now, and this part is about a problem, and this is also why I wanted to have Dr. Priya here. I've made him a captive, so now he has to listen to this. All of our reagents, labor, instrument, and cost pools go into our rates for you. Those cost pools used to be very small. It's now close to $1 million per year that we pay in rent, heat, light, debt service, and stuff. We do get some money from the OVPR. We get about 150 offset of those cost pools, and we get 300 a year for instruments, which we're very grateful for, and that can go to help pay for some of these instruments. Although you can see 4 years of that you'd need to cover that new NovaSeq X. The reason that this matters and that I'm telling you all is that that $400,000 net transfer, it's not from us, it's from you in the rates that you pay. So you're paying money into central funds when you use us. We are not getting funds from central or from your indirects to support you. And I mean, I'm going to probably say this at every GenoFest because I think people don't understand that often and they don't know why we can't do things better, faster, cheaper. And the answer is because we don't get any money from the university to do that. We're not in net terms. So in addition, those cost pools, which include all those things, we've also got like administrative labor and instrument labor above and beyond what is covered. So I and a lot of the other administrative staff that also goes into your rates. And what's odd about this is if you look at the amount of ISO charges that go to the UMGC and that we then pass on to vendors for reagents, the grant money for those would have come with about half as much in indirect. So 5 to 6 million in ISO recharges would have about 2.5 to 3 million in indirect fees, but those do not come to the UMGC, and they don't because we're a thing called a specialized service center. That means that almost uniquely on campus, we're not included in university indirect calculations, which is why we don't get those funds. So when I say UMGC is the MSI of life sciences, I mean it mostly as an extremely positive thing. Genomics, like information technology, has prospered in the last 20 years. It's gone through a couple of phases, and I think, as I hope to argue, it's now poised to just explode. And that's very much like MSI. The state of Minnesota granted a state special in the budget for MSI way back in the 70s, and that's been wonderful for the state. And the fact that your MSI usage is free is because of that. A similar thing has not happened for genomics. There are states that have done it: New York, Connecticut, places in the Bay Area, either from private philanthropy or state funding, have set aside money to support genomics, but that hasn't happened for us. So if you look at the ways in which MSI and the UMGC are similar, we both provide advanced infrastructure. We're both in the OVPR. We both have about 40 to 50 people. They have supercomputers. We have sequencers. We both have softwares and systems to maintain. They get about $10 to $12 million a year. We pay the university $400,000. I personally, over the next few years, I'm going to be spending as much of my time as possible trying to change this at the university and maybe even state levels, because if not, then things like the NovaSeq and all these instruments just won't be coming here to the U, or they won't be coming here and being operated as well as they could. So that's my promise to you all. But the rest of the staff, their focus is on the day-to-day, and hopefully you'll hear about that more today. So thanks. Oh, sorry, one last thing. I was supposed to tell you that we're going to have the first of these new entrants, Singular, who we've actually chatted with, at the beginning of the year, are going to have a seminar on Wednesday, November 9th. Thanks.
Hi, I'm Daryl Gohl, and I lead the Innovation Lab at the UMGC. So just as a brief introduction to who we are and what we do. So the Innovation Lab was established with the goals of, with these 3 goals in mind. So one is helping to catalyze genomics research here at the U of M. So we do that through collaborating with a number of with wide range of labs across the university to apply these genomics tools. Also to elevate the scientific profile of the UMGC, to kind of contribute to some of the methods development and improvements in data quality that Kenny was talking about. And we're doing this through establishing an externally funded program. So a lot of my time is trying to get grants to support this kind of genomics methods development research. And then finally, our goal and mission is to really try to develop methods that are improving the accuracy and reproducibility of the data that we're generating, and expanding the types of measurements that we can make using these DNA sequencing technologies. So this is sort of a little snapshot of some of the things that we've worked on in recent years. We've done a lot of work on microbiome profiling methods and synthetic standards in this field of molecular metrology or measurement science, which is what I'm going to be talking about mostly today. But we've also done a number of projects that are really focused on things that are urgent problems in the state of Minnesota. So helping out with testing and surveillance sequencing for the coronavirus pandemic, working on sequencing the zebra mussel genome and developing ongoing work to develop biocontrol tools. And then also this company that Kenny mentioned, so spinning out some of the microbiome work into Core Biome, which is now Diversigen, which is a company that operates in New Brighton, Minnesota, and generates and analyzes microbiome data, predominantly for kind of industry and pharmaceutical companies. So the microbiome profiling workflow is really kind of similar to like a lot of different genomics workflows in that it has very similar steps. So you take and extract nucleic acids from your sample, turn it into a library, sequence, and analyze. But there are a lot of unique problems that come with trying to sequence a microbiome. So these are complex mixtures of microorganisms which exist. Microorganisms are everywhere in the environment, in the lab. And so, we have to deal with contamination. We have to deal with the complexities of these complex mixtures, which introduce, a lot of challenges in terms of generating, accurately generating libraries, sequencing them, and analyzing the data. And these challenges are really highlighted when you look at studies that look at lab-to-lab variation in microbiome data generation. So this is data from a group called the Microbiome Quality Control Project, where they took a common set of samples and they sent them out to about a dozen different labs. Then they looked at how accurately these labs, or how similar these labs were in their measurements of diversity, or how they sort of agreed or disagreed in terms of how many taxa they're identifying in different types of samples. You can see these measurements are all over the place. Like there's no consistency among the labs. Disturbingly, many of these labs have more taxa identified in their sequencing blanks than they do in the actual samples. So this is hard to do well, and there are many labs out there that are very variable in the results that they're getting. So one solution to this is to design synthetic standards or design controls, reference materials that can be used throughout the sequencing process to control for some of the errors and biases that we know occur during the process. And there are a number of these that have been developed, including these mock microbial communities, which you can use to kind of test DNA extraction and serve as end-to-end process controls. Various standards for various steps of the process, and also benchmarking data sets that allow you to compare the effects of different computational pipelines on data analysis. But one of the issues is that there are a lot of sources of error and bias in these processes that we just don't even know about. And so we can only kind of design these standards for problems that we know exist. So how do we tackle these kind of unknown sources of error and bias? So I'm going to tell you a couple of stories today about how we've uncovered sort of new or unexpected sources of error and bias, either in the microbiome sequencing workflows or in other kind of more generalizable or generalized DNA sequencing workflows, and how we've developed some synthetic standards that let us measure these biases, kind of optimize processes, and ultimately kind of develop tools that we can monitor and control for these sources of bias. When I first arrived at the University of Minnesota, we were just starting to develop amplicon microbiome sequencing pipelines. And so this is the type of data that I was showing you in that earlier slide from the Microbiome Quality Control Project, where it's very hard, different labs are getting wildly different answers. So to develop these pipelines, we used these mock community samples that were made in the context of the Human Microbiome Project. So they made a couple of reference standards that had 20 or genomic DNA from 20 different microbes mixed together either evenly or in sort of like a staggered fashion spanning a few logs of different concentrations. So we use these as ground truth, materials that allowed us to assess the quantitative accuracy of our pipelines. And I won't talk about all of the different optimizations, but through a whole bunch of different, kind of optimizing different aspects of the molecular biology, we developed a protocol that roughly doubled the accuracy of commonly used methods in the field at the time. So the expected values of these community samples are shown by the dashed line here. This is the Earth Microbiome Project method, which is very widely used in the field. And then this is the method that we developed. So you can see we're getting much closer to the expected values. But in the course of this work, we also uncovered a sort of unexpected source of error and bias and unexpected mitigation for that for this source of error and bias. So when you do the 16S amplicon profiling, you're amplifying up a fragment of the 16S gene and the 16S ribosomal RNA gene. And you typically are designing primers in these more conserved regions and then trying to amplify a more variable region that can be used to classify the different microbes. But you can see these conserved regions are not 100% conserved, right? So these so-called universal primers can have mismatches to various taxa. And we can see an example of that here, where within this mock community, there happened to be this organism, Propionibacterium acnes, also known as Cutibacterium acnes. It's this common skin microbe, where there were mismatches between the primer sequence and the 16S template sequence from Propionibacterium, and that caused a dramatic underrepresentation, by many orders of magnitude of this organism in the sequencing data. And so much to our surprise, we found that in certain permutations of the workflow, particularly when we're using proofreading polymerases, which we've been testing because they're more accurate in terms of amplifying DNA, they minimize substitution rates. But particularly when we use these proofreading polymerases, we saw that this organism that was really underrepresented suddenly popped up, and we're suddenly recovering an appreciable amount of it. And what we were able to eventually figure out is that the reason for this is that in addition to being able to correct substitution errors, proofreading polymerases could also chew back the amplification primers and correct them to match the template sequence, and then rescue the amplification of that particular taxa. And if you look at the primer sequences, the sequencing reads from the primer sequences, you can actually see these changes where this mismatch at the second to last position here is now corrected in a subset of the reads to match the Propionibacterium acnes template sequence. So we called this phenomenon primer editing. And then we wanted to learn more about this and develop tools that we could sort of like figure out how this worked in more detail, and then also we could monitor how well it was working sort of on a reaction by reaction basis. So we developed a set of synthetic standards. The design is here. So basically we have a plasmid that has a variable region 4 from E. coli 16S RNA. And we've taken the forward primer template sequence and introduced in 30 different plasmids, we've introduced every possible single base pair mismatch to the primer sequence. These standards also have this second element, which is called a recount PCR-free quantification barcode. This is something I'll talk about more in the second part of the talk, but for right now, this basically gives us a way to really accurately measure the composition of that standard pool. So we can pool these standards together and then measure it using recount, and we see that there's this dashed line here shows the abundance of those standards in this plasmid pool. And then when we amplify it with a proofreading enzyme like kappa HiFi, we get this black curve. So you can see that this primer editing can extend about 5 or 6 bases back into the primer, and then it drops off. So using these standards, we were able to look at a number of different proofreading enzymes. We were able to look at factors like enzyme concentration and how that affected things. Here you can see different enzymes have different capabilities of carrying out this primer editing. We're also able to look at sequence specificity, so we can show there's not really any sequence specificity in which bases get chewed back and replaced to match the template sequence. We're also able to look at or develop a way to tune this primer editing using the incorporation of phosphorothioate bonds into the primers. So these phosphorothioate bonds typically block exonuclease activity, including for different proofreading polymerases that proofreading function of the polymerase. And so by incorporating phosphorothioate bonds at different positions in the primer, we're able to basically dial in exactly how much of this primer editing we want to have occur. And that's important because if you get too much chewback of your primer, you can start to get artifacts off target, like off target amplification or increased primer dimer formation. And so by strategically placing these phosphorothioate bonds, we're able to take advantage of the positive effects of primer editing and the expanded taxa that we can detect. But we're able to minimize these off-target artifacts like primer dimers. And then we can use these standards. So we can basically run these with each of our master mixes and look at, for that particular reaction, how much primer editing occurred. And then we can compare those runs to runs that we've done in the past. So this also provides a tool for us to do sort of process controls and look at run-to-run reproducibility. Okay, so I'm going to switch gears here and talk a little bit about another set of synthetic standards that we developed to measure a property of error and bias that can occur during the sequencing process. So, one of the issues with sequencing in general is that we're generating a lot of our data in these black boxes, and that's literally true in many cases. This is the NextSeq, which is a black box, but it's also figuratively true. These are really complex instruments that have this complex liquid handling system, all this proprietary chemistry that's going on, and imaging and analysis and base calling algorithms that are in many cases they're not open source, right? So to a large extent we're trusting these instruments, right? And we're trusting the QC data that the companies have generated to generate reliable data, right? So this is an attempt to actually look into this black box, right? Not literally like is shown here, but figuratively using these standards. So I've already shown you that PCR can introduce measurement bias, right? And so this is another standard pool or another plasmid pool that we've constructed that has 20 different plasmids that have this little DNA barcode. It's a 20 base pair barcode. So these amplicons that we're sequencing here only differ by this 20 bases of the barcode. And we can see that when we have these 20 different barcodes that we're amplifying, as we do more and more PCR, we get more and more bias, right? More and more deviation from this expected value. So we wanted to develop a method where we could measure our primer pools or our standard pools like those primer editing standards I just talked about really accurately. And so we developed this approach that we called recount, which stands for restriction enzyme enabled counting. And it's really a simple idea. So we basically just take these barcodes and we flank them with Illumina adapters. So there's a sequenceable molecule on this plasmid. And then we can liberate that molecule using a restriction enzyme called MLY1 that precisely cuts out this Illumina adapter flanked barcode. And so we can just take this plasmid pool, cut it with a restriction enzyme, throw it directly on the sequencer. And so essentially what we're doing is just basically counting, we're counting the abundance of the molecules in that plasmid pool. When we do this, we get data that agrees really well with the expected values. So these are 3 different replicates measurements using this recount approach. And we further validated this by comparing to droplet digital PCR data. So droplet digital PCR is a technique that's really gold standard for molecule counting. So we developed these droplet digital assays for each of the barcodes in this plasmid pool, and then looked at the correlation between the recount data and the droplet digital PCR, or between these samples that we'd amplified for different numbers of PCR cycles. You can see we get good correlation with the recount data, but as we do more and more PCR, that correlation is degraded. So we realized that with this technique, we had a way to measure a type of bias that we sort of knew about anecdotally in the sequencing process. So we knew that when we load sequencers, smaller molecules tend to cluster more efficiently than larger molecules. And our sequencing team, Aaron would tell me, "Oh, I'm loading this TN-seq library, which is really small, and you had to really be worried that this library would overcluster on the sequencer, right? And you had to back off on the loading concentration because those are going to cluster much more efficiently." But we didn't really have we didn't really know, to what what the magnitude of that bias was and how it might differ from between different instruments. So we developed these standards that allowed us to measure bias due to sequencing size or due to molecule size on the sequencers. The way these worked is we had one molecule in these standards that had a variable size standard. They range from about 150 to 1500 bases, which is the range of lengths that you can really effectively sequence on an Illumina sequencer. And then these standards also contain a second barcode, which is a normalization standard. These were all a consistent size in every plasmid. And we knew that this normalization barcode was equimolar to its corresponding size standard because they exist on the same plasmid. So when we run this is our first run on a MiSeq. We get data that looks like this, where we see a pretty consistent representation of the normalization barcode, minus how accurately I can pipette these things into a tube. And then we see this sigmoidal curve where we see a really dramatic, orders of magnitude difference between very short fragments and very long fragments in terms of how well they cluster in sequence. And so we're able to run this, many times on various instruments. We actually worked with other cores to run these on different types of sequencers that we don't have here. So we have a very large data set, but I'm just going to simplify it and show you the data for the MiSeq, the NextSeq, and the NovaSeq, which are the kind of three main sequencers that were in production here at the University of Minnesota. And what you can see is that these different sequencers also have very different size bias profiles. So on the MiSeq, we saw that nice sigmoidal curve. The NextSeq had an even more dramatic size bias, so it was kind of more linear. And then the NovaSeq actually had the least size bias of any of these instruments. And so this actually has implications for a number of different types of data that you might generate using these sequencers. So we looked at RNA sequencing data. So an RNA sequencing library is randomly fragmented. And we actually saw, in this case where you're randomly fragmenting your sample, thankfully there is no detectable effect on the quality of the data or on your gene expression counts, because any gene basically has an equal chance of existing in a fragment of any given size. But we did find that there are workflows like ATAC-seq, for instance. So this is a method for measuring the accessibility of chromatin. When you make an ATAC-seq library, you're using a transposon to fragment chromatin, and your resulting library basically looks like a nucleosome ladder. So you get these non-nucleosomal material and then mono, di, trinucleosomes. And so you have information about the biology that's actually encoded in the size structure of the library. And what we found is that when we sequence these ATAC-seq libraries, the same ATAC-seq libraries on different instruments, on either the NextSeq or the HiSeq, which had different size bias profiles, we did see different representation of these different fragments. And that could, in some cases, lead to differences in peak calling when comparing the NextSeq and the HiSeq. So, this indicates that at least for a subset of workflows where there is information about size encoded in the library structure, this can include ATAC-seq or also things like cell-free DNA sequencing, like liquid biopsies, where those are also fragmented chromatin that you're sequencing, basically. This can be an important experimental consideration. You need to take into account, which sequencers you're using. We also found that there are differences in sequence quality that varied by instrument and by size, and that there are laboratory factors. So depending on how these libraries are handled prior to loading them on the sequencer, we could see different size bias profiles. So this is an example where a library was denatured and immediately sequenced, or was denatured, put in the freezer overnight, taken back out and sequenced. And you can see that this dramatically changes the size bias. So aspects of how we're handling the samples and loading the sequencers can also affect this. So we've been trying to now extend on this work and develop kind of a very one really useful synthetic standard that could be used to monitor error and bias in sequencing. Illumina actually uses a PHI-X genomic DNA library right now to report on error rate. So as you're sequencing, you get this dashboard where you can measure error. You can see the real-time reporting of error rate as the sequencing is occurring. So we're trying to incorporate the size bias measurement into PHI-X, and we've been working on generating a standard that we call PHI-Extra. Basically, the way that it works is we've made defined fragments, like we're cloning defined fragments of the PHI-X genome, where we know the size of each fragment, and by the breakpoint, we can tell the size of the fragment. So the breakpoint is basically like a barcode for how large that fragment is. We're then generating these standard pools, and we started to make prototypes that we're now testing and showing that we can basically use these sort of similarly to how you would use PHI-X, but also get this additional measurement of size bias. So, I've told you a couple of things about how identifying some of these, kind of cryptic biases or unknown unknowns in affecting data quality is important for kind of improving the quality of the data that we're generating. And that we can develop synthetic standards. Once we know that these things are occurring, we can start to develop standards that control for them, allow us to measure them, allow us to optimize our workflows in response to these errors and biases. So I'd just like to thank all the people who work with me in the UMGC Innovation Lab and the rest of my colleagues at the University of Minnesota Genomics Center, the many collaborators that we have across the university, and then our funders who have supported this and other work that we've done.
So here we are. I was remembering just a couple of minutes ago, in 2019, I was seated right there, and we were having that discussion, the debate with all the spatialomics vendors, and it blew up out of my mind. It was amazing. And I'm so lucky because here we are 3 years later, and actually I'm part of the spatialomics workflow. And because it's not complex enough, Kenny gave me single cell too, So I'll be focusing today on the standard workflows for spatialomics and single cell. I'm not going to get into all the technical details. There's no time. I'm going to start with the spatialomics because I'm personally involved with it hands-on. It's actually a multi-departmental workflow. I work with Colleen Foster. She's with Bionet, and she actually does the tissue prepping, tissue sectioning. She does some of the stain optimizations. She's very knowledgeable, 30 years. Oh, I shouldn't say that, right? I don't think she's here. She has a lot of experience, actually. It's been great working with her. Work also with Grant Barthel from the Imaging Center. Depending on the workflow, he does the staining of the slides, hybridization, probe hybridization, and imaging, right? Region of interest selection, some of the workflow. John Garbe and I are with the Genomics Center. I'm involved with the library preparation and submit to the sequencing group. And John does the data processing and data handoff. So everything starts with an email, right? We receive an email at our spatialomics@umn.edu. It's usually a question or a request to meet. Most of the time we do end up on a Zoom meeting. Colleen, Brent, and I try to always be present because we bring different expertise to the table. And like Brent, Kenny said, it's not simple. So there's a lot of details to be, detangled, but it's fun. I can tell you that. The two workflows that we work together on is the Visium spatial transcriptomics from 10x Genomics and the Genomics DSP from NanoString. They both are compatible with fresh, frozen, and FFPE samples. They provide whole transcriptome data, protein, a little bit smaller from the panel from the Visium, but it's a larger panel from the Genomics DSP. And none of them provide single cell resolution at this point. You can use single cell data though to kind of deconvolute a little bit and try to get around it and get more information, higher resolution from your data. But those two companies are one of the ones that are going to be launching single cell resolution instruments at the end of this year that Kenny even mentioned. So with the NanoString Genomics DSP, like I said, I'm not going to get into the technical aspect, but this you have the staining and probe hybridization in the beginning of the protocol. Then Grant will set up a meeting with you. He's here, by the way. On Zoom, or you can come in person, and you're going to do the region of interest selection. So after that, if it's a protein project, we're going to count on the encounter. If it's an RNA project, we're going to use the Illumina sequencer to sequence the barcodes, and then you have the data for analysis. I do want to point out that for the Genomics DSP, one of the important parts is the selection of morphology markers. It's not, some of them are very simple actually, and people only use the nuclear stain. There are 3 more channels that are open for other stains, other markers, morphology markers. And you got to think very carefully what your goal is for your experiment, because they help you not only identify the region of interest that you want to zoom in on, but also it actually can be used by the instrument to help capture exactly what's the cell types that you want transcript data from. So something to think carefully. And NanoString has some off-the-shelf, for example, for the oncology group, CD45 and CK is a very common combination, and it's very straightforward and it works really well. Other investigators have custom markers they want to work with, and that takes, I can tell you, adds several weeks, if not months, to the workflow, by the way. But you need to know what you need for your experiment, right? Once your data is ready, John actually then sets up a Zoom meeting, and he'll give you access to the Genomics Digital Spatial Profiler software. You'll have a Zoom meeting with him. He's going to log in. He already runs a QC run, the first step of it, just to make sure there are no like flags or anything weird with your data. And he goes over the software for an hour. That's all you have. And then you need to decide what you're going to do with the data, right? Are you going to work? You can work with MSI-RIS. You can do it yourself, There's a lot of resources. There's the NanoString University. It is a steep learning curve, I'll say, but there's a lot that you can do with the software. And you can export the data actually anytime, any part of the software analysis and do analysis on R and other tools that you might be more comfortable with. So that's kind of nice. NanoString also offers data analysis, by the way. So now changing gears here to 10x Genomics Museum. The difference from Genomics doesn't need a specific instrument for it, but it does have this highly specialized slide with this printed spots that you see. In this case here, this is for the fresh frozen. And what it does, it has those probes that are barcode probes that are going to capture the poly A tail. So it is compatible with any species that you want to use with that has poly A, right, on the mRNA. But they also have kits for FFP samples that are compatible for human and mouse samples. This is a very superficial overview of the workflow. What I wanted to point out is that you have the sample prep, which Colin can help, the imaging part, staining and imaging, barcoding, library construction, followed by sequencing data visualization. In this case, you have more hands-on participation on this workflow. There's a DIY part, which is whatever is happening on the slide, the staining, the cDNA amplification, the hybridization, all that. You're doing it yourself because we are a genomics center. We don't touch tissue and slides. And the imaging center is not feeling comfortable yet with cDNA synthesis, but maybe we'll get there, right? So basically, there's a lot of support. Don't get scared. 10x is wonderful. We set up meetings. They review the protocol. We have a DIY bench at the CRB UMGC lab. We're right next to the imaging center, so it's very convenient. You don't need to buy all those big bottles of reagents that you're not going to use more. You just bring your 10x reagents, and then you're all set. And when you have the data, then John also, well, now it's me, right? It taught me how to release the data. So you use the Space Ranger to do the data analysis and the Loop browser for the data visualization. And you can use single cell data, by the way, to try to deconvolute, as you can see, the number of cells that are in each spot. Same for the genomics. So again, if you need consultation for experimental design, if you're writing a grant and need resources, the vendors have nice packages to support that. We can share that with you. We provide letter of support, quotes. In terms of the project turnaround time, people usually ask us, it's 4 to 8 weeks. It depends if you're going to run a QC run, things like that. But it can be pretty fast if everything is lined up, if the morphology markers are pretty straightforward. Then if you need optimization, like I said, it can take a while. So now, go to the single cell services. So when you contact single cell services, you're contacting mostly is Pat. He is a single cell service manager, but Nasia also might be answering your questions depending on what the question is related to. She is the single cell library prep service manager. We right now, like I said, we're focusing on the standard services. And right now, the only standard service for single cell projects is the next Genomics Chromium single cell. We have a Chromium X series, which is the latest instrument. When you contact via the single cell at umn.edu email, If you're a beginner, usually you're going to end up on a Zoom meeting with Pat and with this 10x genomics support, Egon and Yves-Marie. They're great. They want to make sure that you have your experimental design well thought out. There are a lot of options, so something to be careful about. Think ahead. So this is part of actually the workflow. I wanted to point out here is this part. We're not a cytology lab, right? We're a genomics lab. So you need to become the expert on your cells and tissue. Like all the prep part is the most important part. It is what Kenny said, garbage in, garbage out. This is it. If you have very low viability of cells, what's going to happen is you're probably going to have a lot of ambient RNA, for example, mRNA, and that is going to give you trash data, is going to eat away your sequencing data. So it's very important that you take the time to read on protocols. 10x is very good at giving support on this. And testing. Testing, we can come to the lab, you can ask Pat to take a look at the Luna and look at the cells, number of cells. So it's very important to do that upfront work. And basically, after you come up with your single cell suspensions, it goes through the microfluidic system. It's going to produce the gems, and then the barcode is cDNA. There's going to be a series of QC throughout the workflow. And you're going to have a chance to then take a look at the data and pitch in and say, yes, continue, go to the next step, or no, please stop here. So that's when we do the cDNA is made, you receive traces and quantification. When your final library is ready, you also have a chance to say, no, drop it or go ahead. And you can decide if you want to do a shallow sequencing or jump in directly on an expensive NovaSeq run. With regard to the shallow sequencing, we are reviewing it right now, so it can be more informative. So you should hear from it about it pretty soon. Like I said, there are a lot of options, gene expression alone, kits for immune profiling, multi-omic ATAC with gene expression or ATAC alone. And 10x just released a couple months ago or 3, a kit for nuclei isolation, which seems to be very straightforward and pretty easy to handle. And another one that I wanted to highlight is the fixed RNA profiler that actually Kenny brought up. It's challenging, right? That to come to with your live cells right after you finish, run into the lab, and we can't. That has a cutoff of 4 p.m. because he has to do the downstream part of the workflow, right? And some people then have to start like in the middle of the night, the protocols or something like that, or something goes wrong and slows down, and it causes a lot of stress. So this is actually an opportunity. The fixed RNA brings the opportunity for you to fix your tissue, store it up to 3 months. So it helps with this logistical part of it, but also it can make your experiment cheaper because, scale of economy, right? You can accumulate more samples and then submit them, all of them at once. As with the spatial genomics team, if you need a consultation, if you have questions, we have the email here. It's on our webpage too. They also, are happy to share resources for grant application. Better if earlier than later, like wait until the last minute, guys, be helpful. So we can write letter of support, quotes. The turnaround time target is 8 weeks. Sometimes it's earlier, sometimes it's later. Things don't go smooth, or if people take a while to give the green light, in between QC steps. But yeah, I recommend that we have some resources on our webpage. The UMGC webpage we have a Spatialomics interest group, the C group, that meets monthly. And we have some of those presentations are actually recorded, or there's a video on our webpage, right? And we also have vendor presentations, and that's for single cell too. So take a look, dig in. If you have any questions, if you're not finding something, just send us an email.
Hello, everyone. My name is Aaron Becker. I am the NGS Instrumentation Service Manager for the UMGC. And I wanted to talk to you a little bit today about some of our offerings for our supporting research, I should say, supporting our research with efficiency and value. That's the focus of this talk. I want to basically discuss some of the instrumentation that we currently are offering, and then they're going to cover some pricing issues. This is, I guess, what you would call the Dragnet presentation, which is the just the facts, ma'am, in comparison to all the exciting things that we're we're learning about here. The technology that we're going to see is terrific, but we need to know about the here and now, the daily operations. So first of all, this is the current Illumina sequencer fleet as of summer 2021. So we have the NovaSeq, we had 2 NextSeq 550s, and we have 6 MiSeqs. As of late 2021, we were discovering that the NextSeq 550s had value on the open market. We were getting offers for them, and we decided that it would be a good idea to trade those in or sell one of them to acquire the latest technology. The latest technology for NextSeq, of course, being the NextSeq 2000. So we acquired that, I believe it was around September of 2021, and it's been getting more and more use as we've gone along. The main reason for that is that we needed to have, we wanted to be up to speed with the latest technology, and the NovaSeq, the NextSeq was going to provide us with that sort of next step into the next sequencing chemistry. I just wanted to highlight some of the features of these sequencers. So the NovaSeq is really the workhorse for our high throughput applications. And one of the ways we're trying to realize efficiency for the research community here is to limit the number of runs we're doing and the number of offerings we're doing. So regular runs are limited mostly right now to 2x50 and 2x150 runs on the NovaSeq. That helps us fill flow cells. So we're doing a lot of RNA-seq, whole genome sequencing, metagenomics, and single cell. So what you'll notice is that the NovaSeq is a shared instrument. There are shared runs on multi-lane flow cells. The NextSeq, we're finding that we're using it mostly for faster turnaround times and small to medium-sized projects for researchers who need access to an instrument quickly for a project. The other interesting thing about the NextSeq is that we can get many different run configurations, anywhere from 1x50 to 2x150 right now. And I've heard that there may be an exciting development coming of a 2x300 as well. So we should be seeing that in the future with an upgrade to the chemistry that is going to be introduced on the NovaSeq X or 10. I'm not sure exactly what they're calling it here. So one thing is we're calling it no sharing on this run or on this flow cell, or excuse me, this sequencer. Of course, the MiSeq we're using a lot for our 16S and ITS microbiome work, custom amplicons, and stowaway seq, which is another type of shared run for primarily our microbiome group, but also other sort of test projects. If you need to test out an assay or a sequencing library, you can do it on the MiSeq. Also use it for pre-NovaSeq QC. So I want to discuss some of the NovaSeq options and outputs, and I'm just going to sort of broadly paint these options, not belabor each of the individual options, but you can see there's just a broad range of options. You can have many different configurations, anywhere from a small 100 cycle flow cell to an S4, which is the largest 4-lane 150 paired-end cycle flow cell. Additionally, the outputs are again wide and many. There you can go anywhere from 18 RNA-seq samples at 20 million read depth on one lane to up to 112. So you can see there's a broad range of outputs and yields. So one thing I do want to point out, we're still listing the S2, but it is a special purchase flow cell. We're finding that the S4s are typically outstripping it in terms of cost and efficiency. So we are only offering those as a special purchase. In addition, the 1x100 cycle runs and 2x100 cycle runs on any of the NovaSeq flow cells are special requests, and you'll need to purchase the entire flow cell, which would be 2 lanes in case of those. And finally, I wanted to talk about some NovaSeq pricing. Again, broadly, when you're thinking about NovaSeq, you're thinking about slower turnaround, but a lower cost per read. That is the primary takeaway you should have for the NovaSeq, that it will be somewhat slower because it's shared, but you will see a lower cost per read. And specifically, the current low cost flow cell is the S4 2x150, and it's $2.64 per million reads with our 2023 rates. So it's definitely the lowest cost that we can provide right now. And in the future, with the NovaSeq X, it's going to be hard to imagine how this instrument can compete. So when you're thinking about the NextSeq 2K pricing, you should be thinking about this as your faster turnaround option, but it is it's much more expensive than the NovaSeq. But if you need that flexibility, that would be your option. A little bit about the standard Illumina creation protocols or library preps that we're offering. One of the ways we're trying to realize efficiency again is to reduce the number of different library preps we're offering as standard. It helps us batch library preps. It helps us realize some efficiency that way. In addition, it should allow for lower cost for popular applications we run. So for DNA-seq, when you're talking Illumina Nextera XT, you're thinking metagenomics. For the Illumina DNA prep, we're thinking about whole genomes. And there are other additional applications, such as some legacy Illumina prep, as well as the PCR-free and some low-input DNA options. And for the RNA-seq, we're talking on the top here for Illumina standard input, either mRNA or total RNA assays. So those are the options for those. And then Takara for low input or degraded RNA samples. So what I wanted to move on to here is highlighting one of the services we use to try and help streamline and make our services more efficient. So many of you have heard about Charter Service RNA-seq or used it in the past. So what is it? This is it's a high throughput scheduled monthly RNA-seq run that leverages the value of the NovaSeq high capacity S4 flow cell. So we try and pack as many RNA samples onto a NovaSeq S4 as we can. So the features of this service are: combines library preparation and sequencing for a guaranteed 1-month turnaround. Stranded mRNA and low input are provided in addition to degraded library preparation. So the standard suggestion is 20 million reads per sample, but you can purchase additional depth and 20 million read increments. And one important thing to note is there's a minimum purchase here of 16 samples, and you'll be required to purchase 8 additional after that. And all of this can be had for the low price of $220. So that would include your library prep and the sequencing for each sample. And you can purchase the additional 20 million reads for $64.50 cents, those roundabout numbers. Another way that we are trying to be as efficient as possible is reduce somewhat. I lost my cameras. Is reduce our interactions with you via email and phone calls. And one of the ways we do that is by using Sequencer Home. So many of you may know Sequencer Home. It's a shared Google Doc that we offer. So if you go to the University of Minnesota Genomics website, you'll find up on the right-hand side on the homepage these a toolbar, the tools that we use for sharing this information with you. So we have sequencer queues that are available to monitor your runs, monitor where your project is in our queues. And what I wanted to highlight is the Charter Service RNA queue. So this is a fairly busy chart, or fairly busy queue. What I wanted to point out are the important things that are the monthly date, that monthly run for each of these runs. So you can see on the left-hand side, each of these gray boxes represents the monthly run, starting on the first business day. So 3 important columns of the run date. Secondly, probably the most important part is that you need to know the cutoff date for sample submission. This is 2 weeks prior to the run date. And then third, you want to make sure that you'll know that you should be receiving your data on approximately 1 month after that the run starts. So data deliveries should be 30 days, approximately 30 days following the first business day of the month. And finally, I wanted to just talk about turnaround times here. It's been one of the focus for us as an NGS standard group for the last couple of years, is trying to make sure we're doing things as efficiently as possible and bringing down our turnaround time. So I went back and looked at our projects from the last basically year and 9 months. So, and broke them up into 2021 versus 2022 to see. I had in my mind knowing while I'm processing all these projects that there is a reduction in this time, the turnaround time, but I wasn't sure. I couldn't put a number to it, so I went about trying to do that. So for 2021, we had 271 projects with an average turnaround of 5.4 weeks per project. And in 2022, through actually just yesterday, these numbers go through, we had 239 projects, and we have an average weekly turnaround of 4.8 weeks. So, you know, that's only 4 days. It doesn't seem like a lot, but from the time of post QC to actually generating that data, all the different steps that go into doing that, shaving 4 days off actually feels like it's quite a bit. It's about 11% off of that time frame. So if we can shave another 11% off, actually I'd be pretty happy if we could do that. So I guess that'll be the next focus, is to figure out how we can do that as well. Again, the goal for most projects is about 4 to 6 week turnaround. So we're in that range. So maybe we can go about making that 3 to 5. And I just wanted to acknowledge all the people that make this efficiency possible are listed here. And you're going to hear from Elyse now.
My name is Elyse Froehling. I'm the NGS Service Manager here at the UMGC. Many of you have maybe worked with me before in the realm of standard NGS services, but today I'm going to switch gears a little bit and talk to you about our custom service instead. So as Kenny mentioned, there has been staggering development in the realm of different NGS methodologies in recent years. We are interested in bringing these diverse services to our users, but obviously cannot possibly treat them all as standard and routine preps. Herein lies the designation as custom NGS. This group kind of serves as a catch-all for projects that require some specialized attention. And this really encompasses the more routine but complex preps, such as long-read sequencing and sequence capture, to requests for methodologies that might need optimization or are just brand new and want to be tested out. So today I just want to touch on our repertoire of more common custom offerings, kind of things that we've been working on over the last year, and what we hope to bring to you in the future. So PacBio technology has come a long way in the past few years, and I just want to commend the folks at PacBio for the strides that have been made. We've been really happy with the current performance, and which is a good thing because a lot of the custom work we do here are PacBio preps. A lot of the projects that we take in are whole genome and full-length RNA transcript sequencing, but there's also other relevant applications such as full-length 16S and long-range amplicon as well. In recent months, we've made a few really impactful improvements, namely the conversion to the 3.0 chemistry and implementation of the adjuvant femtopulse, which is allowed for improved interpretation of sample and library quality. So I'll just touch on that briefly before we go forward. So this is a capillary or pulse field capillary electrophoretic platform. And up until late spring of this year, we were attempting to look at PacBio sample quality via the adjuvant genomic DNA tape station assay, which has been a great tool if you want to just look at whether or not the DNA is empirically good or bad, but really doesn't afford us a lot of the sensitivity and granularity we needed to really see what we were working with. So this is the same sample ran on the femtopulse. And as you can see, it provides us with much more insight regarding what the sample and library quality is. And just because this instrument has been so impactful for us, I want to digress for just a moment and mention that while we regularly employ this as part of our PacBio QC, this femtopulse is also an option as part of our standard QC services. And there's also other assays that are supported, namely for RNA and cell-free DNA with detection of picogram per microliter. So how have the updated chemistry and the femtopulse translated into what we're seeing as far as the PacBio run quality is concerned? And we can really look at this in two different metrics that are somewhat related. First being the percent of P1 ZMWs, which tells us really that the library was properly loaded at the right concentration. And then this kind of translates into HiFi yield. So these graphs are just reflect our HiFi whole genome preps over the last year, starting in January 2022. So we kind of generally shoot for about 50% P1 value, and that kind of translates into the expected output of 20 to 35 gigabases per smart cell. And so as you can see, we're on a good trend, and I just kind of want to hammer home what we're seeing in this other trace of the old and new way we're looking at the library and input. This is June. This is the old way and the new way, and we're pretty happy about it. So we're hoping that this trend will continue going forward. So to kind of complement Aaron and our NGS sequencing queue, we also have one for PacBio projects. And so again, you can find it on our website under the tools bar. If you click on the PacBio sequencing queue, you'll be brought to a page that looks a lot like this. And I know it's a little hard to read, but we scroll in, it offers a lot of information regarding project, library type, run type, where you're at in the queue, what the status is. The other long read platform we offer here is Oxford Nanopore GridION. And I really should call it like something like a super long read platform, as there's no theoretical upper limit to the read length. And I believe our in-house instrument has generated a read of something like 1.1 megabases. It has traditionally been used as a DIY option, meaning clients can prepare their own libraries and schedule a time where you can come in and run the instrument on your own. If that's a little daunting, we also offer full service. And I think with any of these long read technologies, you're going to see the biggest benefit by really having high molecular weight DNA as input. So with that, if that is also a little intimidating, we have a very skilled extraction team that has performed this type of isolation previously and has had a lot of success with it. So as far as sequence capture goes, we still continue to see steady interest, and these really fall into two tiers. Some the more standard workflows are off the shelf and readily available. A few vendors that are more common requests for IDT, Agilent, and Twist. There are also other plant panels that require a little bit more customization and maybe a little bit more consultation between researcher and vendor to get it right. And so that is something that we are happy to help facilitate if need be. And also bear in mind that sometimes sequence capture might not be the most appropriate option. And so if that's the case, we're happy to help guide you, and we might actually refer you back to doing long amplicon sequencing. With some of these more highly customized projects, we don't always have a clear outline as to what the project will ultimately entail. In the past, we have supplied researchers with SOWs, which have proved to be a little bit cumbersome. So with this fiscal year, we've implemented a new quoting system that allows for some flexibility while at the same time providing increased transparency, accountability, and consistency. So the quote on the left here is something that you would maybe expect for a standard project where, you know, there are clear deliverables. These are things that we do routinely, and we kind of have a very good idea of what to expect. Alternatively, on the right is one that you would get for a custom project where we still have some catalog services, but now we're able to provide you with a min-max range for custom reagents and also labor estimations. Along with that, the tier of labor. So for example, maybe a researcher 2 would be able to perform the library prep, but you really need a researcher 5 for the consultation and project management. And this ultimately translates then into a min-max range for the total project cost. So beyond the more routine custom workflows previously discussed, I just wanted to touch on a couple of different project types that we worked on over the last year. So we've covered the likes of TNSeq, maximum depth sequencing, and RADseq. We're assisting researchers with custom primer design for long amplicons, and granted a request to do some TCR sequencing as well. We are currently working on a methyl-seq project that's really interesting to us. It involves an enzymatic bisulfite, enzymatic methylation that replaces the bisulfite treatment. This is less harsh on the DNA, and that has ramifications later on for improved sequencing and library quality. This is a really exciting time for us to be able to take part in all of these emerging technologies, and we'd really like to make some of these more readily available here at the UMGC. And I'll just name a few. One being the HiFi prep from Phage Genomics. We have helped facilitate these kind of projects in the past, but in those cases, we've really tried to steer clients to Phage to perform the library prep, who then give us the library for the final sequencing. This relationship has worked really well. We have no complaints, but we're hoping that with additional resources and staff that we can make this part of our custom service fleet. So in the previous slides, I mentioned, the targeted methyl capture as part of our sequence capture option, and NAB's enzymatic methyl-seq for whole genomes. Twist and NAB are now combining forces, and they're coming out with something that it's an enzymatic methyl-seq capture kit. We haven't worked with this yet, obviously, but this is something that's a really exciting new method. So there's also been significant movement, as Kenny and Fernanda kind of touched on, as far as single cell techniques, and also the desire to forego microfluidics altogether. With various approaches, companies like ScaleBio, Parse, and Fluent are looking to streamline this process and make it a more feasible option for standard library processing. And others. So there has been such an immense proliferation of different NGS offerings in recent years as we keep hitting home. But to meet the demands of the time, we are focusing our efforts to make these workflows more attainable to our users. And so with that, we want to hear from you what interests you and what would benefit your research. So please reach out to us at nextgen@umn.edu to discuss your project. I know some scientists would be really happy to chat. Thank you.
I'm John Garbe. I'm the informatics lead at the Genomics Center. Thanks for sticking around. I know you're thinking about lunch, but you're going to want to hang with me here and hear the shocking truth behind our data delivery processes. It actually is not all that shocking, but it's good information to know. So I'm just going to spend 5-10 minutes talking about how we get your data into your hands and what data it is that we're actually delivering to you. We have two data delivery methods. For University of Minnesota customers, we deliver data directly into home directories on the MSI file system. If you are an external customer, you're not affiliated with the University of Minnesota, we have a download website. We post your data to a folder and go to a website and download it. So I'll talk first a little bit about our internal delivery system here. So as I said, we deliver this data right into your group's home directory on the MSI system. So if you log into MSI, on the command line, things look a little different. But if you look at your home directory in home, your group J Smith, there's going to be a folder called data delivery. Inside that folder is a UMGC folder. And inside of that, there are folders for each quarter of the calendar year. Data deliveries are always going to go into the most recent quarter that's listed. So we just started fourth quarter of 2022. So data is now going into that 2022 Q4 folder. The really nice thing about this data delivery folder is the way MSI has this set up. Data in this data delivery folder doesn't count against your storage quota. So if you're at or near using up all your quota space at MSI, us delivering data there is not going to make you hit your quota. We can keep delivering data there. And MSI is covering the cost of storing that data, but just for 1 year. Costs a bit of money to have large data sets sitting around. So MSI has a system where this data is phased out, meaning deleted after 1 year. And the deliveries are organized into these quarterly folders to make it easier to see, well, what's the oldest data that is going to phase out of this 1 year storage time period. MSI will send out emails at the beginning of the quarter that's going to be deleted, saying, hey, you have data here, you're going to want to move it. And then I believe, again, right at the end of the quarter, right before it gets deleted, they'll send out another email. Those emails go out to the PI of the group and to any group admins that the PI is designated on the MSI system. So you're going to want to make sure, pay attention to those emails and take action. What should you do with your data after that 1 year? There's a few suggested options. One of them is to copy that data to another location in your group folder. My recommended spot for you to put that is into the shared folder. Every group has a shared folder. Inside the shared folder is a disaster recovery folder. That's a folder that MSI backs up to tape. That's a good spot to put valuable data like your sequence data. Once you move it over there, it is going to use up some of your storage quota space. If you're full on your quota space, you don't want to buy more, you can copy data over to the tier 2 storage system at MSI. It's less expensive. It doesn't have a snapshotting system. It's not backed up to tape. So it's a slightly riskier option for long-term storage of valuable data. If you have huge amounts of data, which some of our customers do, the university is getting close to launching an archival storage system that'll be available to the university community members. That's as yet not available, but will be a lower cost long-term storage system, location for data. Another great option is to upload your data to a public database, such as NCBI's Sequence Read Archive. That's typically a location where people will upload data when they're publishing results based off of it. NCBI stores your data for free indefinitely. So it's a great spot to push your data. You can always pull it down again. Other collaborators can get it. I believe they allow you to keep it private for 1 year after you upload it. So if you aren't quite ready to publish, you can still put it up there and keep it private for a little while. It's an important note to note that we at the UMGC, we archive a lot of the data we generate, but we do not guarantee that we're going to be able to re-deliver data to you in 2 years from now. So once we've delivered that data to you at MSI, that is the copy of the data. You really need to keep track of that data and have a plan for managing it. If for whatever reason you misplace data a couple years down the line, please contact us. I'll take a look through our data archives and see if that's something we can redeliver, but we don't guarantee that we'll have that data to redeliver for you. This current system of sending data to data delivery has been around for a year. It started just about exactly 1 year ago. Previous to that, we delivered data to a folder in your group directory that was called data release. And that had a UMGC folder inside of that, which had all of your delivered data. The policy for that was that the data was stored there at no cost to you for 5 years. After 5 years, MSI would send you an email letting you know that that data was going to be deleted and that you should move it somewhere else. For external customers who don't have accounts at the Supercomputing Institute, we have a download website, just a web server. External customers each have a folder. We send data to the customer's download folder. There's a few ways to download that data. One is you can load the site up on your web browser, click on individual files and download them that way. Works great for small data sets. You can also download that same data using command line software such as wget or curl. So you can run those commands directly on a server if you're trying to get data from our download website directly onto a remote server somewhere. We also highly encourage people to make use of Globus. Globus is a modern high-performance data transfer software. It's free. There's a little bit of upfront setup, but it's pretty minimal. And once it's set up, all the data we're sending to your download folder, you'll be able to grab and transfer using Globus. It's the best option if you have large data sets or lots of data sets. It will reliably, securely, and as quick as possible move terabytes of data over to where you need it. Data that goes to this download website is available for 30 days, and then we archive it to a lower cost storage system for a year. So you really want to take a look at your data after you download it within that first 30 days, make sure it was downloaded fully successfully. You have a year to come back to us and say, oh, hey, I need another copy, or the download was interrupted. Come and download it again. This option, this external download website, is also available for University of Minnesota customers who don't have an account at MSI. We can just make it available that way, or if you don't want your data going directly to MSI, we can send data this way as well. How to access the data, run wget curl, that kind of thing. We provide instructions in the release email that you'll get notifying you your data is available. Similar release emails go out to internal customers telling you exactly where your data is, how to find it, how to download it. What is it we're delivering to you? For Illumina data sets, we're always going to deliver to you the raw FASTQ files generated by the Illumina demultiplexing software. We never perform any kind of trimming or filtering on the data. You're going to get the data straight out of the Illumina software as it's generated. We run FastQC, which is a quality assessment software that's widely used. We run that on all FASTQ files and provide the FastQC reports to you. And then we also generate our own slightly more comprehensive QC report. Let's see if I can pull this up here. Just pop out of here. Pull up our Chrome. So here's what a basic QC report looks like. We have a plot showing how many reads. What's that? No. Drag it. There we go. Now we can see. All right. So we have plots that'll show you how many reads you have per sample. Plots summarizing the read quality for this is a paired end read data set. So summarizing the read quality of the forward and reverse reads. So on all these plots, the samples are listed across the x-axis. On these plots, we're showing by color the average quality for the length of these 150 base pair reads. So it's really easy to see on these plots. Oh, quality drops at around 100 bases, which is expected for this particular type of amplicon sequence data set. We can pick out a handful of samples that are a little lower quality. We look at adapter content. So how much sequencing adapter are we seeing in these reads? Usually an indication of poor quality for the sample or for the library. How diverse is the library that's being sequenced? How many different variety of molecules were present in that library that went on the sequencer? Oftentimes the absolute diversity level isn't that interesting. You're more looking at individual samples that have a much different diversity metric than the others. That's an indication of potential issues. And then we also take a handful of reads, about 10,000 reads from each sample, and align them against a handful of common genomes and sources of contamination or concern. So we align these all against human, mouse, Phi-X, ribosomal RNA, chloroplast, a couple other things, and just plot what percent of these different references are we seeing in each sample. Since this is an 16S amplicon sample, it makes sense that we're seeing lots of our ribosomal RNA in these. And one handy trick on these plots is on all these plots, all the samples are listed in the same order. And right now they're all listed out in alphabetical order. So this sample here with lots of read depth is the same as the sample that appears right underneath it on all these other plots. And we can change the order of the plots with this little drop-down function. And so, for example, we can go by number of reads, that number of reads. And it reorders all the plots. So again, the samples appear in the same order, but now they're ordered by numbers of reads. So now we can see the curve of you know, how balanced was this library. We can see 4 samples off the end basically have no reads. And scrolling down, you can see those are the ones that also had lower sequencing quality. One of those is the one with higher adapter content. Those are the ones with the 4 abnormal library diversity. So you can kind of help you see correlations in some of these quality metrics across samples. All right, jumping back to, we'll just switch over to the PacBio version. So for PacBio data sets, we also deliver a basic QC report. Most of these plots are generated by PacBio software itself, showing you how many reads were generated, the overall average quality of those reads, the length of those reads. And then if it was a multiplexed run where we demultiplexed the data set for you, we'll have plots here showing how many reads were generated per sample and the average length of those fragments in those individual samples. We also have a little text at the bottom describing exactly what it is we're delivering. Jumping back to presentation mode. I want to point out for PacBio data sets, we are delivering the HiFi reads. So in years past, we would deliver the raw output of the instrument, which is a subreads file. PacBio updated their instruments and their software so that the default output are the HiFi reads. And so now that's the default deliverable we provide to you, a BAM file and a FASTQ file of the HiFi circular consensus reads. If it was a multiplex data set, we'll also demultiplex it and give you individual BAMs and FASTQs of HiFi reads for each sample. We provide the basic QC report. Optional deliverables are, we can give you that raw subreads.bam file, the whole raw run folder. That is needed if you're going to do base modification analysis. So if you want to look at methylation, you need to tell us you want the full subreads data set. That contains the kinetic data that allows software to calculate the methylation analysis. We can also, when we are generating the HiFi reads, we can turn on the base modification analysis mode on there so that you're getting HiFi reads with the base modification data in it. For details about what we deliver from some of our other platforms, like the GridION instrument, single cell, spatial platforms, that type of thing, you can check out our website or you can contact us for more details. And just as a quick update on a refresher on the services we provide at the Genomics Center. Again, we focus on generating the data, not analyzing the data, but we have a handful of services. Some of these are free, like running Cell Ranger, Space Ranger on 10x data sets. Some of these are additional fees, such as GBS analysis, microbiome analysis. Those are run by colleague Trina. The university also has some other resources for bioinformatics support. The Informatics Institute has 2 analysts. Juan Abrante can help you with your RNA-seq data sets or with variant calling. His services are free to University of Minnesota folks. Trevor Gold is the metagenomics analyst expert, can help you with metagenomics projects. For more in-depth projects, we encourage you to talk to the very capable and knowledgeable folks in the risk group at MSI. And I also want to point out that the help desk at MSI can be very helpful in working with you with sharing or distributing your data sets with collaborators. If you need to figure out how do I get my data from my home directory to another collaborator who's going to analyze it, how do I let them get access? But if I have an external customer or a collaborator in Texas, how do I get a copy of the data down to them? The help desk at MSI, they can help you out with some of those data transfer, data access issues. So that covers everything I wanted to talk about, which we're right on time here. Lunch is starting right now out here in the atrium. The presentations are going to resume at 12:30. Illumina will be kicking that off. So come back a couple of minutes early, get a good seat. Enjoy your lunch. Thank you.
All right. How about if we get going? I have the great pleasure to introduce Logan Silber from Illumina. And the reason that I feel privileged to introduce him is because we hired Logan several years ago to work in the UMGC. And like a number of UMGC hires, he graduated from the UMGC to Illumina. So I think we've got the regional sales director, the local sales director, the local FSA, the tech support people. We've got a lot of people doing this. So, Logan, I'm looking forward to hearing what you have to tell us.
Thank you, Kenny. Can everyone hear me all right too? Perfect. So, good afternoon. My name is Logan Silber. I'm a sequencing specialist with Illumina. So, here to support any of you with your library preparation, sequencing, or data analysis needs. As Kenny mentioned, I used to work at the UMGC for about 6 years. So I'm sure for most of you, I've probably actually loaded some of your libraries onto the sequencers in the core. So really learned a lot during this experience and time here. So with that, we're going to talk about a few things today. So first, we're going to talk about a service that is currently offered by the core, which is going to be the Ribo0 microbiome kit. I know from working here that there's a lot of interest in the microbiome as well as 16S sequencing. So for those interested in metatranscriptomics, this is a nice bridge and the next step in getting more insight from your samples here. The second, and as Kenny alluded to, that there were some big announcements at IGF for the Illumina Genomics Forum last week. So I'll be discussing Illumina's new long read technology, as well as the NovaSeq X, which is our newest instrument, which will help to reduce costs even further. So with that, here. So why look at metatranscriptomics? So I know most of you from my time here have done 16S, where you guys are only really interested in perhaps the diversity of it at a species level or a less specific level. And then from there, perhaps moving on to shotgun metagenomics, where you're interested in strain level identification, as well as just knowing what genes are present in the overall microbiome environment. So moving on to metatranscriptomics, similar from DNA to RNA, RNA is going to be what is actually expressed. And from there, you'll be able to understand what organisms are actually contributing the most to some of the biological chemical pathways that are occurring in that sample there. So there are, of course, some challenges with this. So a majority of RNA samples contain rRNA, which is sometimes very difficult to remove. And that can almost be present up to 90% of samples, or at a 90% level for most samples. And then so on top of that, there aren't a lot of very usable metatranscriptomic pipelines out there. And those that are available are a bit costly. So we've had a kit in the past that has utilized or tried to remove the metatranscriptomic or remove RNA from microbiome samples. But we had moved away from that to introduce a new sort of chemistry, which is our Ribo0 plus depletion. We have seen that the Ribo0 plus depletion couldn't quite effectively remove this bacterial rRNA to the same level as the previous iteration. So with that in mind, we want to go back to that level of utility given the interest out in the field. And so we have our Ribo0 plus microbiome kit specifically here. So Illumina has gone about creating a new pro pool that works in tandem with this already established Ribo0 plus kit, which is able to work directly in the workflow and help remove additional RNA from both bacterial as well as human samples here. So also built into the cost of the kit is the ability to access this data analysis pipeline that can be found on Space Sequence Hub. I'll be covering some of the features associated with that. So theoretically, if you were to go through the core, you get your FASTQ files back, you can take those files yourself and run them through this pipeline for free. So as mentioned, we do want to support you from library prep to sequencing to insight from the sample here. So any step of the way for this process, you can always reach out to us to support here. So let's look at some of the quality metrics for this run or for this product here. So on the far left here, we have on these, I guess on the x-axis, let's start there. We have two samples. Initially, if we're going through library prep, we didn't do a Ribo0 depletion. You can see close to 95% of the sample is RNA here. With the orange, you're looking at our Ribo0 plus, our standard kit that's currently out or that is out established. And there's still about 70% of RNA remaining after depletion here. So not quite really where it needs to be. So with the creation of this Ribo0 plus microbiome kit, we've seen that we can really reduce the amount of rRNA to about 20 to 30% here, which is quite good given what is currently on the market too. So this is all been tested against human stool samples. So a lot of utility within the human microbiome here too. So now that we can see what exactly it looks like and how much RNA you can remove to get the reads you need, here's what you can do with it with the data analysis pipeline. So through this app, which is called the Microbiome Metatranscriptomics app, you can get a few features with it. So first, because there's going to be some RNA that we'll get through that we saw, 20 to 30%, it'll actually filter out those reads, and you're left with right here in the green. You guys can see that perfect. The actual reads that you're interested in that aren't RNA there. You'll also get a coronagraph, which gives you a breakdown of what exactly is going to be within this sample here from just a microbial species level here. And then, of course, I think this is actually the coolest part of this app here is going to be the graphical representation of active metabolic pathways in this sample here. So I think you guys can see perhaps, but each of those different colored bars is a breakdown of bacteria and what is contributing to which specific pathway there. So a lot of insight that's, and this is all built into the cost of the kit here too. So, after submitting samples to the workflow, as mentioned, you can log on to BaseSpace and access this app here. So we'll be at the desk. There's some literature on it as well, so we can give you a little bit more of a breakdown. But I just want to make sure that you guys are aware because, even just standing on the desk today, there was a lot of questions around the metagenome. So I'm glad I picked this as a topic for coming in. So with that, what's currently out today that you can access from the core, I'm going to shift gears here and talk about what's going to be coming out in this next year here. So the first topic I'm going to discuss is going to be the long read sequencing. And then from there, I'm going to discuss the X-Leap chemistry as well as the NovaSeq X that I was mentioned before by Kenny. So before we dive into this long read, there's something I do want to make clear with short read sequencing, and that is short read sequencing is very good. There is a FDA precision challenge that was put on back in 2020, where a variety of different sequencing companies put forth their sequencing data as well as created different, and also use different data analysis pipelines out there to essentially see what is the most accurate when marking against either benchmark regions or also specific regions, such as the MHC region, which I'll get to in a second here. So with improvements that we've made with this Dragon 4.0, and for those who might not be familiar with Illumina's secondary analysis pipelines, this is something that we're constantly continuing to update and improve, which is why over here you can see that we're tied between other long read companies in terms of the accuracy when it comes to the comprehensiveness of the human genome. So we're at 99.83 with short read sequencing here. So short read sequencing, very good. That is what I just want to say there. And then also when it comes to even some of these specific difficult regions, such as the MHC region, we see that it does come out a bit ahead in terms of other long read sequencing. So you get the read depth that you need with short read sequencing, and also this the associated cost. So this helps to just show that we can cover a large majority of the genome. But with that being said, there are still portions of the human genome that are just difficult with short read sequencing. So with that in mind, this is what kind of led the idea behind this new library prep for long read sequencing. So the Illumina Complete Long Read Prep, which previously you might have heard that was called Infinity before. So these are the same items or assays here, is a highly accurate, comprehensive genome and adds to our current short read sequencing. So as we saw before, we were at 83, and now we're sitting at 99.87. So this is a great complement to our current short reads to help improve accuracy here. So you can see here the Illumina Complete Long Reads bumped up there. This is a library prep and data analysis solution, so no new instrumentation is needed for this. So you can load this currently on the NovaSeq and NovaSeq NovaSeq 6000 and NovaSeq X. In terms of throughput needed, we are going to recommend high throughput just to maintain cost effectiveness. But at the end of the day, this is going to be a very simple workflow with a 50 nanogram input and about 10 kb size that we're looking for the insert, or at least not insert, but the input. And also a simple workflow, and that this is a one-day workflow, and we are seeing that it can technically be automated here. So we're really trying to streamline this process for human long read sequencing here. So just to kind of give an idea too of the overall full workflow, you don't need any specific special extraction equipment here. There's no real stringent requirements in that sense. We've tested exactly some of the limits of the input, and we're recommending 50 nanograms of input as far as our qualifications there. We have tested down to 10 nanograms of input and also seen success there. But once again, we want to just guarantee the 50 nanograms. So no shearing required in that sense. This will be high molecular weight stuff at the end of the day. But we really wanted to strive to improve this workflow. And using this instrument, you could actually get up to 10,000 human genomes through our enrichment method, which I'll get into. And then also if you're doing the long read prep, that'll be 3,000. So just to dive in that. Oh, yeah, and also as mentioned, built into the cost of the kit is actually going to be the analysis for this application here. So Illumina is definitely trying to ensure that when you purchase a kit or go with one of our applications, you're going to be supported from start to finish there. So as I was alluding to before, there's going to be, you can fit onto the NovaSeq X Plus here up to 3,000 high accuracy genomes a year, which is quite a bit more than some of the other ones on the market. And we're looking for this assay here about at N50s of 6 to 7 kb, and reads can possibly be generated up to 30 kb here in this case. So really trying to ensure that we're covering that last 5%, 5 to 7% of the dark genome, which short read sequencing has issues with generally. I'll be covering some slides here shortly showing how short reads compare to long reads for this new Illumina assay. So if we're turning to HLA coverage, which is or HLA genes in specific, pretty common gene that does have some difficulty and has a lot of utility out in the clinical space. You can see that short reads, while covering a good portion of this region here, the 7.3 kb region, there is this region highlighted here that starts to tail off and just does have these issues. Compared then with our Illumina complete long reads here, we do get that coverage that we're looking for. So like due to the low coverage of the short read sequencing, we're not calling some of the variants that we do see called in this long read here. So if you look closely, and I can share these slides afterwards, you can see that there are 2 haplotypes that are being called now because of the Illumina complete long reads. So let's take a look at human trios now and how the benefits of using a complete long reads in this case here. So these sample trios are part of a publicly available 1,000 genome project, and they've identified a Puerto Rican family trio here. So while zooming out and looking at the Illumina complete long reads, and all these are Illumina complete long reads with the father, mother, and child, you can see that there's a deletion here with the father, a homozygous deletion in the father, but not in the mother. And then you can see with the child here that there's actually going to be a heterozygous deletion here. So comparing that to and just honing in that the father of this trio here, you can see how this compares once again to other on-market long reads and quite quite good coverage here. So something to note too with this assay too is that we're currently and looking to continue to improve the data analysis pipelines, as this is going to be a large portion of what carries this workflow here. So this is only just going to get better and better in terms of comprehensiveness and accuracy as the product continues. So what I've discussed so far has been complete long read sequencing. It'll be a library prep that's complementary to our short read genomes here, covers the whole genome. And just to be transparent, there'll be quite a bit more coverage needed for this workflow. So that's why we found that this actual library prep is compatible with enrichment. So with enrichment, we're hoping to take a panel, perhaps a panel, for example, that covers that 5 to 7% of the human genome and be able to just match those regions across the short read region to help get the most cost-effective and comprehensive human genome out there on the market. And you can kind of see here too, I'll move into this. So at the GBA region, here's the Illumina short reads once again. You can see that we just lose coverage completely over here. Here's the Illumina complete long reads. You're getting the full coverage here. And then here's us with our capture for this specific region. And you can see that we cover it very well. So something else, and just to show further data here, just showing the efficacy of short reads versus long reads, that this is definitely compatible with enrichment. Something else to note, this will be compatible with custom panels as well. There's a roadmap for that later on. So we know there's interest in, say, for example, pharmacogenomics and understanding those regions, as well as MHC regions themselves. So you can have these specific portions if you're only focused on them and focus on those applications too. If there are also specific regions that are less than some of the ones I've spoken about, that should be compatible, but will be in the future. one other added benefit before I move on is going to be that you can understand phasing too. You can get phasing information directly from this Infinity protocol using the data analysis pipeline. So very clearly defining between the allele 1 and allele 2 here. So just in terms of timeline, as you think about your projects moving forward for human long-read sequencing, at the beginning of this next year in quarter 1 here, so probably around February, we'll see that this whole complete long-read assay will come out. And then later on, the second half of this year, you'll have the enrichment capabilities coming along with it. So stay tuned for that. And I'm sure there'll be some questions here afterwards. I'm always happy to answer them too. So with that, as I mentioned, there's going to be a lot of need for read depth. And so with that in mind, it wasn't a coincidence that we've helped to develop a new instrument that will increase throughput even further and also reduce costs. So NovaSeq X is the culmination of our announcement of Chemistry X, which has looked to improve accuracy, reduce the time of sequencing runs themselves, and then also help reduce costs. So this is a rework of the SPS chemistry that everyone here is very familiar with, what they've used an Illumina sequencer before. This isn't just one new thing that we've added to the workflow to make this work. It's been a rework of creating new molecules, improving the hardware itself, adding Dragon servers or Dragon FPGA chips to the instrument itself to improve analysis as well. Something too that for me, working at the core, and I used to have to unpack some of the reagents here, and that can sometimes take up to half a day. With this chemistry, it's actually stable enough to be shipped at room temperature. So no more dry ice, no more ice packs, a lot more compact in what is being shipped for you for one run. So also looking forward for sustainability in your sequencing runs, we kept that in mind when improving this chemistry here. So with that, let's see how it compares to the NovaSeq 6000. Or actually, sorry, we're getting a little ahead of ourselves there. Let's talk about some of the flow cell configurations with this instrument. So at launch, at the beginning of the next year, it'll be the 10 billion flow cell. This is going to be comparable to the NovaSeq S4 flow cell. One thing you might notice from some of these images here is that the 1.5 billion flow cell, which is going to be comparable to an S1 NovaSeq 6000 flow cell, there are 2 addressable lanes. For the 10 billion and the 25 billion, there are going to be 8 addressable lanes here. So going back to the HiSeq, and this will also assist in making sure that you can fill out a run in a cost-effective way for your lab as well. Some of the runs or flow cell configurations are also shown here with 100, 200, 300 cycles for the 1.5, and 10 billion, and 100, 200, 300 for the 10 billion, and then 300 for the 25 billion. And I just want to also focus on that 25 billion, which, as Kenny alluded to, is almost 3 times the output here. The NovaSeq 6000 with this flow cell. So as we move forward here with this instrument, there'll be 2 addressable flow cell lanes with this NovaSeq X Plus. So you'll be able to have 16 possible projects going at the same time, perhaps here. So how does this compare though? With these improvements, we're actually still able to reduce the speed of the runs. If we're thinking about a NovaSeq S4 run, which only costs or takes about 44 hours to run, with this new chemistry, we're looking at about 24 hours here. So same output, but reduce the cost, these 8 addressable lanes here. In terms of output, if we're talking about the amount of human genomes, just to give some context, with the NovaSeq 6000, you're utilizing both sides, you're looking at about 48 full human genomes, versus now with the 25 billion flow cell configuration, you're looking at over 128 or 128 human genomes here. So, which also adds up to about 2.5 genomes an hour. We've come a very long way from the Human Genome Project, which took years just to get one. So really impressive to see how far things have come here. So just in terms of accuracy and how it compares to the current SBS chemistry, I just want to show some metrics that we're seeing on our side. In terms of phasing and error rate, and also incorporation time, we've been able to reduce all these metrics across the board here by about 50%. So from standard SBS to XPS to this new XLEAP SPS chemistry, you can see that we're really just trying to improve things the entire way through. And this is also the first iteration of XLEAP chemistry. So once again, continue to move things along. So all these improvements are nice, but what does that mean for the lab and as far as cost goes? We're seeing that with this chemistry here, you can get about $2 per gigabase moving forward. So some of the metrics being thrown around here are $200 to $250 per human genome. So if we're looking at also just cost per gigabase compared to the NovaSeq here, with the S4 flow cell, it's about $4.9 per gigabase. Comparing that same flow cell configuration and output to the NovaSeq X 10 billion, that's going to be $3.2 per gigabase. I saw there was mentioned too in some of the slides as far as the services offered, the cost per million reads of the NovaSeq 6000. The cost per million reads with the 25 billion is going to be under $1, probably close to about 80 cents there. So we're really pushing it here. To give some further context about what this can mean for barriers of entry and the reduction here, here's how much it costs now for a whole genome to be sequenced versus a whole exome sequencing. $102 for whole exome sequencing. And you can see the majority of the cost is going to be coming from library prep versus $285 for library prep and sequencing, both with Illumina assays on an Illumina instrument. As Kenny alluded to as well, there are Dragon servers or Dragon FPGA chips on board, so that can assist with demultiplexing as the run goes on, as well as pre-built secondary data analysis pipelines that you could utilize. So data analysis is also built into this cost here if you're going with those specific pipelines in that sense. So really, once again, trying to make this as accessible to people as possible. I do really want to emphasize once again the 8 addressable lanes, just because if we're going from the 4 lanes of the NovaSeq, where if you're talking about an S4, that's 2.5 billion reads or million reads that you would need to fill out for your project. Because we're not going by 8, you can think of that as almost more 1.25, which makes it a little bit more manageable. That's going to be more associated with the NextSeq 2000 P3 flow cell. So you're still in that high throughput range, but very accessible. It makes it a lot easier for you guys to access the cost savings of the $2 per gigabase here. So also one of the other benefits with the NovaSeq, for doing a standard run, you'd probably need a decent amount of volume and material to be loaded across the flow cells themselves. But with these 8 lanes here, it's about the same input as an XP flow cell, or sorry, XP loading concentration. So you're talking maybe 18 to 22 to 40 microliters of starting input there if post library prep. Coming then at the beginning of 2024, this chemistry, the XLEAP, is going to be coming to our mid-throughput system, the NextSeq 1000, 2000. So a P4 flow cell configuration will be available. So just once again, to make it clear that we're continuing to improve upon not just our data analysis tools, but also the sequences that are currently out there. And if someone were to go with an instrument, we're not just going to move away from the chemistry. We're bringing also that XLEAP chemistry to the previous flow cell configurations as well, P1, P2, P3, to help reduce costs in that way. So we'll get more information on that, of course, as the year goes on. But first half of 2024 is what to look for this flow cell. And this, of course, as mentioned before, you can't share a lane with the NextSeq, so this can also assist for those who, if they don't want to wait for other lanes to be filled, could be a great option for also reducing costs in a mid-throughput setting. So just to summarize here some of the benefits, you can do a lot more with less now. So you don't need as much input. The speed and the amount of materials, I removed some of the slides showing just the size of the cartridges and reagents, but just a reduction across the board in terms of even the size of the flow cell that can still get almost up to 2.5 times the same reads as the 10 billion here. So reductions across the board in the size and the waste produced here. From just the return on investment, you're looking at $2 per gigabase. So the best cost savings per gigabase out there on the market, hands down. And once again, I'm just going to talk about this because I love that it's shipped at an ambient temperature, no dry ice. I think it's just a great improvement to the work of life quality within a lab too. So that's everything about the NovaSeq. Well, there's a lot more I could talk about the NovaSeq X, so I'm looking forward to questions. But before we go to questions, something I do want to plug. Coming soon, Illumina will be working with the University of Minnesota Genomics Center on a pilot project grant program. We'll be announcing some details about that at the beginning of next year, but essentially, if you have a project that you're working on and you need sequencing reads to help supplement a application you have for a grant, you could apply for this and perhaps qualify for funding for some sequencing materials or library preps here. So stay tuned for that. And if you have questions about your experimental scope or needs, feel free to reach out to me, and I'll also get the booth afterwards too. So thank you.
Afternoon, everybody. How's everybody doing? Great. My name is Chris Black. Some of you may know me from various other manifestations, other roles, other organizations I've been with in the past. Today, I'm here as a representative for PacBio based in the Midwest. My family and I live out in Madison, and I'm really excited to talk to you about our technology and chiefly about some of the applications for that technology. So I'm going to start with a technology overview, and I'm going to warn you, I'm kind of a nerd about molecular biology and genomics technology in general. So if this turns into a black hole for me and I like fall down the rabbit hole and talk about this for an hour, my friend Matt is going to wave at me and make sure that doesn't happen and possibly come push me out of the way. So warning. Okay, so in general, the way the HiFi sequencing workflow happens, we start with high molecular weight DNA that's derived from any sample type that you choose to work with: viral, plant, animal, human, microbial. And we ask you to isolate high molecular weight, high quality DNA from that sample of choice that can then be used to generate high molecular weight, long individual input molecules. So single molecules of double-stranded DNA. You can then see that we ligate these specific hairpin-shaped adapters. Those adapters help us generate what we call a smart bell library. And I'm going to touch a little bit on the unique characteristics of the smart bell library in that they are single molecules that are structurally linear but topologically circular. We're going to talk a little bit more about what that means in a second and why that's important. From there, we can actually bind a polymerase to the bell portion of that library molecule using a specific primer. And once that primer and polymerase are bound, you'll start getting integration events around that molecule, that single molecule that's in the smart bell configuration. From there, we actually load those molecules onto a chip that has very small wells built onto it. The complexes are captured in those smart wells, excuse me. Ideally, one and only one of those complexes per well. And then the sequencing occurs on the Sequel II platform that's in-house here at UMGC. Okay, so I wanted to show just a couple more images, dive into this slightly more granularly. You can see up at the top left, we have a picture of the smart chip. This is where the actual sequencing occurs. You can see the sort of golden hue on the bottom. That's actually a CCD chip that's capturing images in real time, not cyclically, but as a continuous movie. So we're actually capturing an image constantly of integration events and the fluorescent signals that are produced therefrom. So we load single molecules into these tiny wells. We capture the image from inside that well. When fluorescently tagged nucleotides are integrated, we get a signal, which you can see depicted down here on the bottom. And it's important to note that we're actually getting two key pieces of information. We're getting signal base calls from the movie that's captured. And we're also getting what we call the interphase duration. So you can see these arrows here. Normal integration events have a very predictable length of duration between them. You can see that sometimes we get a longer phase of duration between those integration events. That's actually the case when we have a methylated base that's part of the input molecule. So using those kinetics, we can look not only at the base that's being integrated, but the methylation state of the base that's being integrated as well. All of this in conjunction allows us to build high consensus accuracy through multiple circular passes around that structurally linear but topologically circular molecule. So I keep using that terminology. What does that actually mean? Here's a graphical depiction of what we're starting with. High molecular weight double-strand DNA. You can see that the, for lack of a better term, plus strand is depicted in orange and the minus strand is depicted in purple. We ligate our adapters, creating the smart bell. We add our primer and polymerase and start the synthesis reaction. And then the cool part of all of this is we actually have a circular molecule where we can travel around the circle through multiple passes, around and around and around the circle. We're sequencing the entire length of the positive strand through the adapters, the entire length of the negative strand through the adapters, and back around and around and again. The reason this is important is because, as I mentioned earlier, we are actually working with a single molecule here. And almost any single molecule technology will inherently have difficulty with signal to noise ratio issues. You just don't have a lot of stuff there from which to generate signal and parse out the noise in the background. What that leads to is relatively high rate of random errors. You can see here in the depiction, the red nucleotides are actually erroneous calls. These are calls that were mismatched, that were not accurate. By passing multiple times through that molecule, because these are random errors, we can build a consensus accuracy that's incredibly high, and we can filter out those random non-systemic errors. So passing around the molecule multiple different times means that we can build to an incredibly high accuracy, long HiFi read down here at the bottom, which is depicted as our green read. Okay, so we do the multiple passes. We're actually leveraging the fact that we have over 100 kb potential total base pair reading, but we're doing that circularly to get molecules that are HiFi as consensus that are generally on the order of 15 to 25 kb. That's kind of a sweet spot for genome applications, somewhere on the order of multiple kb for some other applications. What that generates is all of these amazing claims that I'm making over here on the right-hand side: extraordinarily high accuracy, even coverage without systemic biases, complete coverage including dark regions of the genome, allele level resolution and long-range phasing, the best performance for all variant classes combined. I'm making very bold claims here, and I'm not going to show a bunch of bar graphs and stuff to back that up. What I'm going to instead ask is, if you think I'm being hyperbolic, please give me a call and let me show you the data sets, show you the publications, the comparisons, all the other stuff that includes all the variant types, structural variants, copy number variations, single nucleotide variants, etc. All that stuff is available. I just don't want to spend all of my time talking about it, because otherwise we'll be here until tonight. Okay, I want to shift now and stop talking about the technology, although I love the technology, and start talking about the applications, the way people are actually utilizing this technology right now. So the first example I want to give is actually de novo assembly. And it might seem weird for me to be talking about de novo assembly and showing human genome telomere to telomere and human pan-genome reference consortia, because we know that the human genome has been available for quite some time. Mostly, I want to call out that two of the lead PIs that are associated with these two consortia have said really amazing things about the value of having high fidelity long reads as part of their consortial efforts. And I want to dive a little bit more deeply into each of these and kind of show what they've been working on and why we think it's really exciting and important. So the first here is the telomere to telomere consortium. So everybody raise your hand if you think the human genome was sequenced about 20 years ago. Nobody? What if we had said that 6 months ago? Everybody's hand would have been up. So the first reported whole human genome sequence was sequenced about 20 years ago. This was a huge event. All of the work that Kenny highlighted earlier in his talk. What that did not include are a number of regions that you can actually see depicted here in black. They're kind of hard to see because there's so much. Frequently centromeric regions, often regions that are segmentally duplicated or that have high numbers of repeats. And in fact, there was about 8% of the total genome that had not been previously sequenced and successfully mapped. The telomere to telomere consortium was a large, diverse group of researchers that came together and said, we're going to finish that 8% of the genome. And they used a number of different technologies to do that. In fact, everybody that's here today that has a booth outside probably contributed in one way or another to the telomere to telomere effort. We were really proud to see that there were quotes like the one here at the bottom coming out about the value of HiFi reads and actually building the complete telomere to telomere sequence of the entire human genome. This was published in Science in March 31st of this year. So the first human genome that was completely sequenced came out in March of this year. Not 20 years ago. None of you guys fell for that. That wasn't fun at all. In parallel, there's another group who's working on building a human pan-genome reference. This is a consortium that has funding from the NIH to sequence at least 350 whole genomes, human whole genomes that can be combined together to produce a human pan-genome reference. So rather than going back and saying, I'm going to look at the human build GCRH 38 or whatever, 37 that I'm working on, this will be a combination of different genomes that can be used for reference analysis simultaneously as a pan-genome. And one of the primary directives of this group is to look at historically underrepresented genome populations and make sure that the entirety of the diversity of the human population is represented in this pangenome set. There was a publication from earlier this year where they published the first set of those 350. It's over 40 human whole genome assemblies. And again, you can see down on the bottom that there were innovations in PacBio HiFi sequencing that allowed all of this work to really be done. And of course, it's not just humans that we want to do de novo assembly for. There are a host of species, all of these species that are important for agrogenomics work, for biodiversity efforts, and for model organism research, where we can also use native long reads in the current manifestation of our technology to build the highest quality reference genomes possible. As you can imagine from the description of the human pangenome reference, sometimes de novo assembly is not what you want to do. You actually want to refer back to a human genome that's already been characterized. This is amazing work from the folks at Kansas City Children's Mercy Hospital, where they looked at over 1,000 pediatric rare disease genome cases. These are sometimes called diagnostic odyssey cases. A kid comes in and has issues. We don't know why, what's going on, let's figure it out. They went back to over 1,000 samples that they've sequenced in the past and used HiFi sequencing to try to understand, looking simply at structural variants, how many more diagnoses could be made using just structural variant information. And by simply incorporating structural variant information, you can see that over 13% of new explanations can occur that were previously unsolved using short read technologies. Now, obviously, those cases are really relatively rare. We think of these as rare diseases. But there are other manifestations that can benefit from this technology as well. Here's an example of using structural variant analysis in childhood cancers. This comes from the group at Nationwide Children's Hospital that are looking for these very complex rearrangements in genes that are associated with childhood cancers. This is the retinoblastoma gene. You can see, we know that cancer is a disease of the genome. This genome is completely shredded to bits and rearranged and inverted and all kinds of terrible, crazy things. And this actually, if you go back and look at this publication, led to the discovery of a novel fusion transcript between retinoblastoma and this SIAH3 gene as well. So lots of interesting stuff that's being done on the structural variant side of things using whole genome sequencing. The other side of this is looking at those medically relevant, challenging, dark regions of the genome, where there are a number of genes that are represented here that generally have evolved as a result of some sort of segmental duplication, right? So you have a gene that's absolutely vital to the survival of an organism. Evolution comes along and says, I'm going to duplicate that so that I have the room to make other cool new proteins and do other cool new things. And in so doing, ends up with two pseudogenes that can be incredibly similar to one another. So greater than or equal to 90% identity. What that leads to is a very difficult time mapping reads that do not span beyond those regions of high homology. Using high quality long reads, we can actually span those regions and we can start integrating more information about those dark regions of the genome. Another class of variants that historically has been really challenging to work on are what are called variable number tandem repeat variants, or VNTRs. Here's a really interesting example where the WDR7 locus, the gene that's, or a gene that's known to be associated with ALS or Lou Gehrig's disease, can be tracked evolutionarily. And we can start to see both at an evolutionary level, how many of the repeats are expected to be present in these various different species, but also within individuals. This is a pedigree of a family that's known to be familially affected with Lou Gehrig's disease. And you can see using, in this case, a long-range PCR and amplicon-based approach, that we can look at the variable number of tandem repeats that are present in various individuals of this family. We've actually just released a brand new bioinformatics tool that allows us to look at VNTRs called Tandem Repeat Genome Tool, or Target, and that's available from PacBio now as well. Yet another area that's significantly of interest to a number of researchers is pharmacogenomics. The pharmacogenomics genes, 50 of which are listed as part of a panel that has been developed in partnership with our friends over at Twist. You should talk to those guys when you're outside later. These genes are very similar to one another, very difficult to map. A whole host of them are a member of this superfamily called cytochrome P450 genes. These are largely involved in the metabolism of different drugs, which is why we care about them in terms of a pharmacogenomics standpoint, the way the drugs are metabolized and utilized by your body. And you can see here examples of us being able to differentiate between CYP2D6, which metabolizes something like 25% of all of the drugs that are currently given to human beings, and CYP2D7, which are approximately very close to one another and have a high degree of homology. Awesome. That is fantastic. I love it. Thank you, John. The fact that we're working from single input molecules and we're capturing these long reads from those single input molecules means that we get full phasing information. So you can see here shown in IGV, the haplotypes that are captured from the maternally derived and paternally derived alleles. In pharmacogenomics, this is particularly important because it means that we can get star level annotation of those two alleles for both of the alleles that are present. Okay, CPGs. Let's talk a little bit about the epigenome and being able to use that interphase, interpulse duration to look at methylation events that are happening in the genome. I'm showing an example here from Matt Bainbridge, where they're looking at Prader-Willi syndrome. Prader-Willi syndrome is kind of a canonical example of where imprinting matters and where different epigenetic changes can actually really matter. The depiction here at the top shows both genes are maternally derived and hypermethylated, means that they are shut down. This is someone who's afflicted with Prader-Willi syndrome, and we can see that from the epigenetic reads that are shown here on this plot. Whereas the controls, you can see, have one hypomethylated and one hypermethylated copy of this particular gene, meaning that they're actually able to produce the protein product and avoid the consequences of this terrible disease. Similarly, this is an internal example. I apologize, it's somewhat difficult to see the IGV plot down on the bottom, but we're looking at a particular region where we can use imprinting and phasing simultaneously to actually look based on the epigenetic profile of this region and see the maternally derived and paternally derived alleles of this particular locus. Beyond that, we can also look at the other side of epigenetics, which is looking at parts of the genome that are accessible and parts of the genome that are inaccessible using a technique called FiberSeq. This was originally described by the STAM lab out of University of Washington. And essentially the idea here is, rather than using something like a transposase, which would be used in a TAC-seq to assess which part of the genome is accessible to enzymes, they use a methyltransferase. The benefit of that methyltransferase is that it's very, very small and it's able to get into the nucleotides and around the nucleosomes and actually change A residues to methyl A residues in an observable manner at a single fiber level. So along any one strand, along any one copy of the genome, we can actually get methylation data that indicates whether that part of the genome was accessible or inaccessible to those methyltransferases. What that means is that we no longer have to look at chromatin accessibility in a bulk manner. We can actually look at individual molecules and understand if there are different changes to the accessibility along those strands that allow them to be transcribed or turned on or turned off or shut down, et cetera. And some of this work is being conducted right now in a way that's allowing researchers to start to elucidate the specific mechanisms by which those epigenetic modulations are being made, and then translated into biologically relevant wrapping within the genome itself. Okay, switching gears again, I want to talk about IsoSeq. IsoSeq is the idea that rather than looking at only a tag from a gene at its transcriptional level, we can look at the entire isoform of that transcript. So we can go from the 3' start all the way down to the 5' and look at the entire content of that gene as it's being expressed. There's some really amazing work that was published by a team in Singapore where they use this technique to look for alternative stop sites in genes that are known to be important for gastric cancers. We're showing one example here where if you look at the Kaplan-Meier plot, the line plot that's down here on the bottom left, you can see that the known promoter produces a gene product with an insignificant difference in survival between high expressors and low expressors. So if I make the normal gene, it doesn't matter if I have a lot of it or a little of it, I will have roughly the same outcome from these gastric cancers. Alternatively, they found a non-canonical start site for the same gene that truncates the protein and whose expression is significantly different, whose Kaplan-Meier survivability curves are significantly different based on high and low expression. So it's not just the fact that this protein is present, and it's not just the fact that the protein is present in a high or a low level. It's the idea that you have a different protein that's actually present based on the start site of the coding. Beyond that, they also looked for all sorts of different classes of canonically interesting iso-seq splice variants, where you can look at different types of splicing that indicates the overall gene product. I also want to highlight another area where iso-seq is particularly interesting, and this is looking at single cells. So we can actually do iso-seq on single cells. And in this case, we're looking at very precious human brain samples. So you guys might be surprised. There's not a lot of people that are willing to give up a chunk of their brain so somebody can come in and try to understand at the transcriptional level what's happening in those different cells. We actually have research here that's showing that by looking at the specific isoforms that are present in different cell populations, as well as in different disease states, we can understand at a deeper level what's going on in those very precious brain samples. Further, there is a very new sort of nascent technique called MOS iso-seq, which allows us to take the best advantage possible of the long reads that are possible on the SQL platform. So we've talked a number of times about having 15 to 25 KB of usable space that can be generated within each one of those wells that's on the chip. Most transcripts are on the order of about 1 KB, so you can see we're not very efficiently using that space. MOS iso-seq is a mechanism of concatemerization where we can put together multiple isoforms into a single read and then read all of those isoforms together. What this means is that we can read about 15 to 16 full-length transcripts per molecule and get more and more isoform level information from the same Chromium libraries that are currently being generated on 10x. So you don't have to change your single cell workflow. You just stop at the level of full-length cDNA production and feed these into the MOS iso-seq. We're expecting to have a kitted version of this likely within the next couple of weeks. I didn't say that. Long read sequencing, high quality long read sequencing is also incredibly valuable when you're looking at complex populations like you would have in a microbial setting. So we are looking here at shotgun metagenomics. And in fact, this is HiFi data that's combined with our friends from Phase. Is Phase here right now? Phase is outside. You should go talk to Phase about how high-seed data can be combined with long reads to produce the highest accuracy possible metagenome assembled genomes. Essentially, the idea of that is we have a very complex mixture of all kinds of microbes, simultaneously sequence all of them, and build complete closed circular genomes for those bacteria in that complex sample without having to isolate them and look at them independently. I am using up all of my time, so I'm actually going to very quickly simply mention that we have an assay for whole genome SARS-CoV-2 analysis that uses molecular inversion probe technology. It's very interesting. You should come chat with me about it if you have any interest in SARS-CoV-2. We can use this for patient-derived samples. We also have a version of this that is in late phase development for wastewater monitoring for whole genome of SARS-CoV-2 in wastewater. And we have excellent performance of that assay. I'm going to ask a couple of things of everybody that's here today. I'm going to stand outside at the booth with the pretty pink banner. Please come by, talk to me, introduce yourself. tell me that you loved what I said or hated it or thought I spoke too fast or that I'm flippant or whatever you want to say to me. I don't mind. Just come by and talk to me. Ask me for proof sets. Ask me for evidence of the very bold claims that I've made, because I have done exactly that. And we do have the proof to be able to back up what I've said today. Happy to provide that. When you're thinking about your next experimental design, think about HiFi. Think about how to use long reads. You're going to do a bunch of Illumina sequencing, and that's fantastic. I love it. Think about how we can augment that sequencing and what we can provide that may be additional and supplemental to that. I'm going to put my email address and my colleague Matt, who's sitting over here, so that you guys can reach out to us. Everybody grab your phone and take a picture of that so you know where to reach me. I appreciate literally everybody doing that right now. Thank you. And keep working closely with these guys. They're amazing. They utilize the technologies that they have in an incredibly skillful way, and they enable research that couldn't be done otherwise. We genuinely really appreciate everything that you guys do. Finally, everybody be well. Take care of one another. Be kind. Thanks. I probably have time for a couple of questions. Brandon, whenever you're ready, come up here and kick me off the stage, man. Kenny, what's up?
You didn't mention the short read sequencing.
I know I didn't mention the short read sequencing at all, and it's incredibly interesting. So my expectation is that by the early part of next year, we will have a lot more information to share about short read sequencing, our sequencing by binding technology, which is unique. Offers incredibly high quality reads. There's a little conference at the end of this month called ASHG that a lot of people sometimes go to. It's entirely possible that we would have information about SPB at ASHG. Any other questions? Thank you all very much. I genuinely appreciate it. Cheers.